Good morning to everybody. I would like just to take a few seconds to, on behalf of Scuola Normale Superiore, to welcome all of you and to open by this welcome this symposium on changing brains, neuroplasticity, neurodegeneration, and neurodevelopment, neuroprotection, in honor and memory of Luciano Domenici, a great scientist, a great person, and a great human being, and a friend. I would like in particular to welcome our guests from outside of Pisa and from overseas, because this is a sign of great appreciation of what Luciano did in science and in relationships. And together with Alessandro Cellerino, Nicoletta Berardi, and Alessandro Sale, when thinking of this event, we thought that the natural way to remember Luciano would be to hold a scientific event on the themes that kept Luciano enthusiastic for his scientific life, busy for his scientific life, and to which he gave huge contributions. And also, we thought that it would have been natural to host it as a joint event between Scuola Normale and Institute of Neuroscience of National Research Council, that is a natural partner of the neuroscience at Scuola Normale throughout decades due to the common origins with Lamberto Maffei. And without keeping you more, I thank you very much for coming, and I wish a good and productive day. Of course, there will be space for personal memories in the afternoon session, and this is also a very important and probably a very important part of this meeting. And I leave the mic to Alessandro Sale for...
Thank you very much. I speak on behalf of the Neuroscience Institute of CNR, and of course today I'm touched because we are meeting together to remember a great person, which was also a friend of us, a friend of our institute, the Neuroscience Institute of CNR. Since the very beginning, I have to say, since the late 80s, when Luciano won a competition for a position of research scientist, independent research scientist in our institute, that in that period was directed by Professor Lamberto Maffei. But later on, he continued to remain in tight contact with us, for instance, as senior research scientist, and also with important, relevant positions such as that of director, scientific director for a while, or member of the scientific council of our institute. And I think that Luciano is a real example of a passion for life, passion for science, passion for research and teaching. And also when this passion for teaching led him to leave the Institute of CNR to get the position as a full professor of physiology at L'Aquila University, also in that period it remained in tight contact with us, with many of us, for instance maintaining his laboratory in Pisa and also continuing a number of collaborations with many of our colleagues. There is an ancient African proverb that states that when an old man dies, a library burns to the ground. And so I think this proverb is correct. So today we are sad. We are sad because we lost this library. We lost this friend. But I think if I look that we are so many today, that there are reasons to be also glad. We can be glad because Luciano is an example, and if we decide to follow that example of passion for life, passion for science, well, I think that a part of that fire, that light, which was the Luciano's light, can continue in our example, for instance, in our students, in juveniles, in the passion for science and research. So thank you all for being here once again, and once again a special thanks to our speakers who accepted the invitation and are here to share their thoughts and their work with us. So have a nice meeting. I leave the microphone to...
Prima Michela, vero? Prima Michela. Ok. I will chair this morning's session, and so I will introduce the first speaker, which is Professor Michela Fagiolini from Harvard Children's Hospital. I will share with you my memories of Luciano this afternoon, and so I will just say that I am particularly struck by the picture that has been put on the leaflet, because that's Luciano's smile. He always had this smile lurking in his eyes while speaking, and he was clever in making terrible puns and flashlightning short sentences which made us laugh. And so I particularly like that picture, because it's that my friend, which I remember, and whom I met for 10 years every day because we shared the same room. Those who know me and know Luciano might wonder how he survived, because he was very tidy, very precise, and I am very untidy, and he was silent, and I am a chatterbox, but he put up with this for 10 years, and so I'm glad that we shall proceed to this through this day under those smiling eyes. So, Michela.
I think I have the mic.
Okay.
Okay. So first I would like to say thank you. Thank you for inviting me. What? I turn it on. Do I have to bring up? Okay. Is it okay now? Okay. So I would like to thank the organizer and friends, actually, for organizing this symposium. It was when... Okay. When Alessandro asked, I was like, yeah, that's good. That's the right things to do. And I should not be the first. But Luciano was not a friend. For me, he was a mentor. It was the first person who taught me how to drill a cranial window on a mouse, on a rat. At the time, I had rats. And he was always the first to ride to the lab before everybody else, no matter what, if he was healthy or not healthy. And always the last to leave. And that is an example when you're a young student, barely knowing where the brain starts or finishes, to know that you can do if you want. So, because I don't want to cry through the entire talk, I just want to say thank you. And thank you, Luciano, because you will be with us always. And I always mention Luciano when I talk to my students or to the people who come and say, you know, it's difficult to do science. I know there are no position, there is no money. How can I do? I know in Italy it's difficult. And I say, you can do, you just need to want to do. And then you do, there is no point. Yeah, you will not be rich, most likely. You will not have a stable position, even when you're 50, even when people call you professor. But you do, because you love what you do. And then you will be successful, because successful is measured not on the number of publications, but if you can change the life of someone. Okay, let's try to start to work. Okay, thank you. So today I will try to tell you what I've been doing for the past, now roughly 10 years in Boston. I work at Boston Children's Hospital, Harvard Medical Schools, and after a journey that started here at the Scuola Normale and the Institute of CNR with Lamberto, Luciano, Nicoletta, and everybody, where I study plasticity of the visual system. Then I work little briefly in San Francisco, moved to Japan, and end up in Boston. And there I decided to study neurodevelopmental disorders. And the idea, the interest was everybody's different. We are who we are as adults as a result of the interaction between genes and environment, genes that we receive from our parents, the environment that we are embedded. But sometimes these trajectories, this beautiful process that brings a baby unable to speak several languages or just barely Pisano, sometimes you have insult, and insult can be at the level of your genes or the environment where you are, and that trajectory deviates from what we call normal. There is nothing normal, but let's say neurotypical, and given set of neuromental disorders. Because I'm very not creative, I decided to continue to use the system that I learned here, the visual system, and use the visual system as an entry point to understand the complexity that give rise to disorders. The visual system is developed in an experience-dependent manner, so babies are born, can see, but their vision will mature in the few years after life. We can measure behaviorally by just making the babies preferentially look at bars compared to gray screens, and increase the spatial frequency, making the stimulus more difficult. You will see that the baby will not eventually distinguish left and right, and you can measure what we call visual acuity. And this happened during the first year of life when neuromental disorders happen. I don't study humans really. My patients are animals. I study rodents for the entire my life. I started with rats, and I thought they were stinky. Then I moved to mice, and they are really stinky. However, they are good tools for science because they still have 2 eyes. Their visual system is organized pretty much like in humans: 2 eyes, 2 thalamus, a cortex, the input convergent LGN, and then a right to visual cortex. They don't see very well. They're nocturnal animal, but yet they do see, and their vision actually can be measured behaviorally with the task that was developed a few years ago by Dr. Prusky. And their vision is very poor at eye opening, so they are born with their eyes closed, they open around P12, but very quickly in the next 2 weeks they will mature and stay stable. And so the idea was, can we use this to measure vision in animal models of disorders? And at the beginning, when we established the lab, we decided to try to use this optomotor task of visual evoked potential and measure what we call optomotor acuity. And we measure in several lines of mice that are model of disorders of autism, in bread line like the BTBR, and insult, pharmacological insult like the VPA, that if happen in humans, actually women exposed to VPA during pregnancy have a higher risk to have a child developing autism, or monogenic, like the MCP2, that is a disorder, gene that when mutated, give rise to Rett syndrome, or a protein like the neuroligand 3 that is a synaptic protein that is involved in the establishing of excitatory inhibitory connection. All of these mice, you can see the white bars is a control, the shaded or black line, bars are the mutant, are different from the controls, suggesting that there are some defects, even in primary sensory modality. We focus on the MCP2 monogenic line because it was the one that gave us the strongest defect and was the line that allowed us a better control of what's happening. We know the genes, we can manipulate the gene in specific circuit, and so maybe it's an easier entry point for understanding why this defect arise. So what is Rett syndrome? Rett syndrome, as I said, is a neurodevelopmental disorders, is rare disorder affecting 1 in 10,000, 15,000 new births. It's a gene that is expressed on the X chromosome, and for that reason, boys actually die at birth or soon after. So it's essential for the survival of the human being. In mice, the mice, even male, will be born and survive, so it's less critical, but yet they will die after 2 months. In a girl, because girls have a 2X chromosome and they have an X activation, they are mosaic. So they're in the brain and in the rest of the body, they have cells that they do have a normal MCP2 and other cells that lack MCP2 or have a mutated form of MCP2. This mosaic is sufficient to allow an apparent normal development, initial development for the first 6 to 18 months of life. And the babies reach milestone, able to speak or say a few words, use their hands, communicate, move, maybe walk even, and at one point they stop progressing, and at that time is when the parents start to notice abnormality, something's wrong, and the diagnosis arrive. And the diagnosis, more than being for the mutation of MCP2, is given if the girls undergo regression. So when they hit stagnation, they start to lose in the next year or so the skill that they have acquired and do not acquire new skills. During this regression, there is a presence of repetitive hand movement, a very stereotyped movement, a problem in gait. Often the girls eventually end up on wheelchairs, delay in cognition, and onset of seizures and respiratory abnormalities. At this moment, we don't have a cure. Although we know that a mouse can be treated, if you introduce the gene, a mouse that was going to die will survive, will come back and look good. However, we only are able to help the quality of life of this patient. We don't have a successful treatment, we don't have a cure. We can cure mice. If you are a mouse with Rett syndrome, you are lucky, you can be treated. But our goal is not to do that, is to arrive and treat them. So, what does MCP2 do? Lots of stuff, and MCP2 is a methyl-binding protein. It's been studied extensively in the epigenetic field, and Adrian Bird actually has elucidated the major role of MCP2, that is a suppressor. So it works in a complex, bind to the DNA to particular island, the CPG island, and prevent gene expression. Now, over the years, the roles of MCP2 have been expanded. Many studies have shown that actually could be considered an activator, has a role in chromatin compaction, and so on. But the bottom line is that it affects gene expression. And this expression affects gene expression of genes that are responsible for the formation and maturation of the brain. As I said, you can reverse the defect, and this gave hope, and this is not degenerative disorder. The cells are still there, the brain is suffering, the cells are smaller, they have lost their dendrites, but they are spines, but they're still there. And again, if you reintroduce the gene, you will recover some aspect of that. So going back to us, what we found at the beginning, when I show you at the beginning, I show you the acuity of the mouse with the optomotor here, when they were fully symptomatic and adult. And so what we found by taking advantage of this task that does not require any training of the mouse, we were able to show that actually vision, this optomotor acuity, was developing and then regressing, very much in line to the disorders where you have, as I said, a normal, quote, normal development and then a regression. And so that actually was the result that put me in business at the beginning of my lab, because now I had an assay that could be probed and also an entry point to understand why this is happening. The previous slide was done in male mice, total knockout, but when we use also female, the heterozygote model that is closer to the patient because it's a female, and as I said, the mosaic, yet developing the mice, the phenotype of Rett syndrome appears later. We use visual evoked potential, for example, and we found that even the female do have a visual impairment. Their visual response to low spatial frequency is reduced, so the amplitude is smaller than in a littermates wild type, and their acuity is decreased, very much like in the knockout. So that brought to the question, okay, this is a mouse, what's about in the patient? At the time, there was not so much known about the patient other than they use their eyes to communicate with the parents, but nothing specifically about how well do they see. And because the patients are non-verbal, it's very difficult to have a classical evaluation of perception because they're not able to communicate really what they are seeing. So in collaboration with the Rett Syndrome Clinic at Boston Children's Hospital and Chuck Nelson, we decided to pretty much do exactly what we have done in the mice. Although we didn't anesthetize the patient, although we didn't do a craniotomy, we just placed these nets, EEG nets, on the skull of the patient and then record visual evoked potential in response to a very simple checkerboard, alternating checkerboard that elicit a very stereotyped response that appears in the first year of life. So it was, let's see what we get, and are we able to actually get some response in these difficult patients, these patients on the wheelchairs, often they grind their teeth, often they may have a seizures during the recording. So because of that, was not a given that we will be able to record VEP from them. But we try, and if you try and you don't give up, you are successful. We said, let's try. We had to exclude the sum of the traces, maybe repeat the presentation a few more times than a neurotypical subject, but we were able to get a response, and we found, this is already published, so I don't want to go too much in the details, that indeed the red curve comes from a group of 34 patients with age between 2 and 12. They do have a visual response, but the response in amplitude is decreased compared to the control, and the morphology of this response is also abnormal, suggesting that the intracortical, either top-down, feed-forward, the processing is not exactly the same as in the neurotypical subject. And this is quite important for us. Also the acuity, we measure in a subset of patients the acuity, so when you increase the spatial frequency, so the checkerboards become smaller and smaller. Again, this is really the first pass. We can do much more sophisticated testing, but the result was very much in line to what we have found with the mice. These patients do see less. Worse, when the stimulus become difficult, they are unable to detect it. And so we can also use these nets to look what happened at the level of the entire brain. And so this is a meta-analysis that we did after, that is actually now ongoing, where instead of looking just in the visual cortex, we look at the signal throughout all the other electrodes. Because this is a very simple stimulus with the checkerboard, the response should be exclusively in the visual cortex and very robust. Red means signal, blue means no signal. And as you can see in these two other panels, here are the patients, we resorted them between active regression, so they're younger patient, closer to the 2 years of age, or post-regression. This division was done on the basis of the clinical evaluation. You do see that the response progressively decrease, and also the response is not localized as much as you see in the neurotypical subject. Again, giving us some hint of what's going on in the brain. Right now, the evoked potential, and not just the visual, auditory evoked potential gives similar result. Visual seems maybe a little more reliable across multiple sites, meaning right now evoked potential being recorded not just at Boston Children's Hospital, but in Texas, in Colorado, at other Rett clinic. And they're part of the natural history study for Rett syndrome and related disorders, with the idea that maybe we can use evoked potential as a biomarker for the disorder, for the progression of the disorder, and maybe one day even as a biomarker for outcome from clinical trial. Right now, the clinical trial that have been conducted for Rett syndrome all failed, but actually across many neuromental disorders. And often there is the criticism, well, you found something in the mouse by testing something that is totally unrelated to what's going on in the human brain. And maybe for that reason, when you do this big jump, you fail. You need higher mammals or closer mammals to study the disorder. Maybe yes, but at the same time, maybe no. Maybe if you use something that is more sensitive, less biased by personal judgment. Many of the clinical trials run with a questionnaire filled by the primary caregiver, and the emotional component is huge. So maybe with this quantitative unbiased readout, you have a better chance to see a result, positive or negative. And so going back to the lab, we wanted to understand why this happened. Where does this defect arise first? What are the mechanisms, the cellular mechanisms underlying the dysfunction, and can we rescue? So I will try to touch briefly into these three questions. Some work is being published. Most of them is not, as usual. My lab is quite slow in publication. But the first thing was where does it arise? We know the visual system is start from the retina up to the cortex, and all of these stations, like from the retina to the geniculate, from the geniculate to the cortex, are subject of plasticity and experience-dependent refinement. And around 2012-11, my colleague Qin-Fei Chen showed that actually the retinogeniculate circuit also are abnormal in the mice lacking MCP2. At the same time, we showed that instead in the cortex there is defect. So where is this defect that we see in the cortex comes from a defect that is driven by the thalamus or vice versa? In fact, the retina, thalamus, and cortex are interconnected. Usually we think in a feed-forward manner, but we know that there are input coming back from the cortex down to the thalamus. And this input that we have shown in neurotypical developing mice can impact the refinement of this connectivity by manipulating the feedback. And so what we did, we used an animal model that has an extensive time for developing the defect, and we look at the retinogeniculate circuit, both in slice preparation and also in vivo with single unit recording, and an age when the animals are still asymptomatic, before we do see a major regression. And in the thalamus, we did not see any significant abnormalities. I'm not saying that there is zero abnormalities, but we were unable to detect. And instead, when we move the electrode from the thalamus to the cortex and record again the evoked potential or single unit response in the anesthetized animal across all the layers, we saw that in the animal lacking MCP2, at this young age, P30, both the spontaneous activity and the evoked activity of pyramidal excitatory neurons was significantly decreased. Already there was a loss of activation, a severe impairment. It will become worse. So it worsens with time. So if you look at these mice when they are 2 months of age, then the silence is even worse. But it's already present very early on. And so first conclusion is probably a feedback that drives the regression of the thalamus. We are trying to publish this now, but brought us to focus more on the cortex and say, why do you have this silencing? Are the excitatory cells losing already their ability to spike, or because the cortex is an integration, it's a circuit, it's not just excitatory cells, it's not just inhibitory cells, they're all interconnected with each other. Could it be that inhibition actually is stronger and contribute to the silence and the worsening of the activity of the excitatory cells? Of course, the answer is yes, otherwise I will not say this hypothesis. And what we did was with the single unit recorder, we record excitatory inhibitory cells and then compute a ratio, excitation inhibition, the old idea of excitatory inhibitory balance that many years ago we put forward with Takao. And we found that in the wild type mice, you see that this ratio change with development. So excitation does not change so much, but inhibition increase and the ratio decrease over time with maturation. But in the mutant mice, this ratio is already shifted in favor of inhibition. And in fact, if you look at the gamma power, you see that the gamma power is increased. The gamma power is generated by inhibitory cells, in particular large basket parvalbumin cells. And you see that there is an increase very early on in the 30 to 55 and 12 to 30 hertz. 12 to 30 is probably mediated more from somatostatin, but this is generated definitely from parvalbumin cells. So we have conducted some anatomical studies. These large basket cells make synapses on the soma of pyramidal neurons, and they are able to control and direct the spiking of pyramidal neurons. So we did an immunostaining. This is a picture of P60, where we compare wild type and knockout with staining PV and GAD65 and count all these numbers, all these puncta. And the result is the following, that even at P15, you see that the number of PV pancreas on these pyramidal cells is increased. There is a gain of function of these cells that persist throughout life. And so you can start to think that because there is this hyperconnectivity, maybe is a contribution in the silencing and unbalancing of the circuit function. We look more recently at other inhibitory neurons to see if the PV were just one of the many that are changing and which one was changing first. Here I show carotene and calbinin in addition to parvalbumin. We look also at somatostat and VIP cells, and the paper is just being published, but we find that while PV is already abnormal at P15, the other inhibitory circuit either are stable or they start to change later on, almost like a compensatory event. In fact, if you look at the adult and you look at a general marker like GAD65 or 67, you will see a decrease of such markers, like inhibition is suppressed. But we do think that is not all inhibition is the same. And so there is, as I said, a progressive imbalance. So can we target this circuit and rescue the defect? The PV circuit mature in an experience-dependent manner postnatal. In the visual cortex, if you keep an animal in the darkness, their PV circuit will not mature. You're exposed to light, they will mature. And so we start to look specifically at the input, excitatory input onto the PV cells. And again, this work has been published, but we found that there was again, earlier maturation of the excitatory drive onto PV cells. And indeed, in the human, there were already reports in post-mortem tissue from patients with Rett syndrome that there is an abnormality, an increase in an NMDA expression receptor. At this point, in this tissue, we don't know which subunit is misregulated. In the mice, we know that the subunit glue R2A and 2B are shifted in their expression. So the PV cells express more 2A very early on, while pyramidal neurons will maintain a higher expression of 2B, remaining more immature the pyramidal neurons, instead more mature the excitatory cells. We have done some we have done some electrophysiology in vitro, and array tomography to prove that. We have done some genetic manipulation of these subunits, and that was enough to rescue to some degree the defect of Rett syndrome. And again, genetically is not an easy intervention for the patient, so we start to think about pharmacological manipulation of these receptors. And we decide to test the ketamine drug as the first compound, not because ketamine is a particularly clean drug, but more because it's an FDA-approved drug that is already regularly used in the clinics, even in children, because there is a rapid effect for sedation and you can metabolize and wake up quickly. We know from work done in the past in vitro that at low concentration, ketamine acts preferentially on an NMDA receptor expressed on PV cells. And so with these two ideas, we start to think, can we use as a treatment? This is not really linear because this work was done after we tested in vivo, but I just wanted to, we wanted to really be sure that this treatment was specific for the receptors on PV cells. And indeed, what we did was to test the impact of ketamine on activation of the pyramidal neurons. So what I told you before, the MCP2 mice have a silent cortex. So what we want to do is to reactivate, remove that silence. So we would like to increase activation. When you give a ketamine, if ketamine acts onto the PV cells, and block the NMDA receptor, you will block the activation of the PV cells. So inhibition is prevented, and there is a disinhibitory effect. In fact, if you record the baseline activity of single unit, you record the baseline, and then you give ketamine, and then you record again after 30 minutes the activation of the pyramidal cells, the very same cells will start to spike more, indicating a disinhibition. And you see that there is an engagement of gamma sort of gamma power, sort of. If you do the same experiment, but in this time in animals that lack the subunit 2A, GLUN2A, one of the two subunits that together with the NR1 form the NMDA receptors, what you see is that while the little male wild type have the disinhibition, the increase in activity, the mice lacking 2A are unable to upregulate their activation. And in fact, even in the spectrogram, we don't see any change before and after. But again, this is a knockout where 2A is lacking everywhere, in every single cells of the brain. And so we use a conditional animal where we remove selectively 2A onto PV cells. And what we found was, although not complete, there was a decrease, significant decrease in the upregulation, the disinhibitory effect that we see with ketamine, strongly indicating that a low dosage of ketamine, this is 8 milligram per kilogram, actually is sufficient to block the activation of the receptor and have a disinhibitory effect. So, as I said, this is a little bit backward because this paper we published in 2015 or 16, where we used that concentration and administered not acutely but chronically every single day in the mice ketamine, with the idea that if we can slow down or prevent the maturation inhibitory circuit, maybe we will have an impact on the outcome for Rett syndrome. And so we started either a P15 very early on, because we know that PV cells are already hyperconnected at the time, and a P30. And P30 was very important because that's the time when the regression starts, and in a patient, that's when you will start any clinical trial. Long story short, the treatment was successful. The PV circuit will be still miswired when you give a vehicle to the animal, but will have fewer puncta on pyramidal cells when you give ketamine. But the more important part was that these animals not only were able to see better, their cortex was more active, but they will not die. They will not survive as a wild type that never dies, but will not die at P60 like a vehicle-treated animal. The quality of their life was extended, and there was an improvement even in their respiratory function, in their coordination. And this is a boost because it means that what we study in the visual system may be expanded to the rest of the brain. Not all the circuits are made the same, but it seems they are not maybe so different. Indeed, right now there is a clinical trial ongoing for ketamine. And I think these results are part of the reason. However, we were not so supportive of going into clinical trial right away because it's still a mouse. The mice still die. And you have to give ketamine. Ketamine is a drug that is, when taken a little bit too much, hallucinogenic, or anyway, give you hallucination, okay? So it's a good drug, but you have to take it with granosalis, okay? And also this study was done in the male, and I said that the patients are mosaic female. So we started to do a sort of clinical trial in female and use the VEP as our outcome, and we got some encouraging result, but not as strong as we wanted. So the amplitude of the VEP is still reduced compared to a wild type, and is intermediate between a ceiling-treated and, as I said, the control. Also, the general health of the mice is not so strong. And so we start to think that maybe the female were not responding exactly the same as male. And this is work ongoing. We don't have the final answer, but we went back to the recording in vivo and tried to see, are the female responding to this acute injection of ketamine exactly like the male? And the response was like, sort of. Was very variable, and we didn't see so much increase in the vocal or spontaneous activity after the acute injection. Why? We start to think, why? Well, male, female, well, oh, maybe the Estrus cycle, this brilliant idea, could have an impact. And indeed, when we start to sort our females, these are females that are already have reached puberty and going through estrus and the estrus regular cycle every 4 days. And we start to make a vaginal swap at the end of the recording and sort the data. You start to see that females that are in estrus, so the red part and this one, will not respond to the ketamine. But females that are not in estrus behave like a male. Opening yet another question, why? And so there is actually, this is published, and this is all done in wild-type mice for now, and now we are doing with the MCP2 female. But what we found is that the gene expression for the NMDA subunits actually is modulated in PV cells, is modulated by estrus. I'm not saying that it's not modulated in other cell type. We have just started, of course, from the PV cells and pyramidal neurons, because are the one where we see that we care for. But definitely, you have less expression of 2A when you are in estrus, and more expression similar to a male when you are not in estrus. So this complicates the work. And so before jumping again from the mouse, the human, we should know also in the human, but we have to be careful. So that's why I'm saying you cannot jump so quickly. You need more work. It's more complicated than people would like to have. But I think we are getting there. And now we are doing the preclinical study and trying different concentration and so on. But hopefully you will see. So this brings me to the last part of the talk, and if it's too long, I stop, where, as I told you, I tried to convince you that the PV cells seems to be experiencing a gain of function in the absence of MCP2. But fortunately, we know, thanks to the work of many, many people, especially Takao, but many people here as well, what are the factors that control the maturation of these cells. And we know that there are both cell autonomous and non-cell autonomous factors. And so at one point we decided, can we start to think about non-cell autonomous mechanism? And in particular, okay, so this is just to tell you that the PV circuits actually are hypermature throughout the brain, not just in the visual cortex, and we have used also tissue from patient and typically developing control, look at motor and visual cortex, and confirm that there is a hypercomplexity and maturation in PV circuit in the subject, the human subject as well. And these cells indeed have a much more complex phenotype. So not just their axon is more elaborated, but also their dendrites. And this work was done with another person from here, Graziella Di Cristo, who prepared at the now is in San Giustin Hospital, and we collaborate with her, and she works with organotypic light sculpture, so she's able to feel the cells and reconstruct them fully. You see that the PV cells are very, very elaborated and complex and hypermature. So although their soma size is smaller, so like all the cells in the brain of Rett patient or MCP2 mice, that there is a suffering of the soma, their arbors are more complex. And again, we are looking in the postmortem tissue, and with DAB is not as beautiful as an immunostaining, but seems to give a similar result. And so factors that contribute to the maturation of these cells, one of these is this very interesting and funky protein OTX2. OTX2 is an homoprotein that is important for the formation of the brain. So if you don't have OTX2 early on in development, you basically lack the brain, but they get re-expressed in the choroid plexus and in the periphery like the retina postnatally. And Takao, in collaboration with Alan Prochance, have shown that actually OTX2, the uptake of OTX2 by specifically of PV cells is sufficient to trigger their maturation and open ocular dominance plasticity. So we thought that, oh, maybe we could tap on that mechanism and see if we can slow down again the maturation of PV cells. Why that? Because if we can do that, we could access OTX2 in the choroid. The choroid is much more accessible because it's in the ventricle. You can do intrathecal injection and maybe gene expression. So it was sort of intriguing and will affect the entire brain. So we did immunostaining first for OTX2. Is OTX2 in the brain in the absence of MCP2? And the answer was, indeed it is increased both in visual cortex and in motor cortex. And there are more cells containing OTX2 already at P15. And often they colocalize with the PV cells. You can try to disrupt the uptake of OTX2 using peptide. Again, is a trick that was developed for the ocular dominant plasticity. We know that there is a particular sequence, a peptide, that allows OTX2 to be picked up and internalized by PV cells. So we can interfere with endogenous OTX2 by infusing either RK peptide that will compete with endogenous or a control AA peptide. And what we found was that by doing such injection intracortically, we were able to rescue the overexpression of PV protein and the connectivity, and also the intrinsic properties of these cells. These cells are fast spiking cells. In the case of the mutant, they spike even faster. And when we use the RK peptide, they go closer to a wild type distribution. So then we went and said, okay, how is OTX2 in the choroid? We did immunostaining, and we found that both choroid and retina had more OTX2 than the wild type. And especially in the choroids, the increase of the protein for OTX2 stay elevated throughout from P15 to 60. And then again, we try to say, what about the patient? And it's not that easy to have access to choroid tissue. However, we were lucky to have access to cerebral spinal fluid from neurotypical subject with meningitis, and a small group, 10 patients with Rett syndrome. And how did we get that? Well, the controls were patients who came to the Boston Children's Hospital because had a fever, and with no cause. So the first things they do a spinal tap, and they take 10 cc of cerebral spinal fluid. Then they run some tests, and then they keep the vials for a month, up to 3 months in the refrigerator until the case is closed, and then they discard. However, you can ask and say, can I have that vials? Maybe 2 years ago, those vials, some vials, the permission to get the vials, but eventually we got. And then you can start to resort those vials according if the patient had developed a meningitis or other infection or nothing to be detected, and that's what constitutes neurotypical. You can also ask to brain bank, indeed, for cerebral spinal fluid. Together, we were able to see that these controls had either a low level, if they were neurotypical, or an increase with the meningitis. However, the Rett patient also had an elevated OTX2. And these subjects were collected because the very first clinical trial for Rett syndrome was done for IGF-1 treatment. Was a study that was published by Daniela Tropea, another student here with Miranka Sur, where they showed a treatment with IGF was sufficient to help the reduction of the phenotype of Rett syndrome in mice. And so because IGF is again an FDA approved, a clinical trial was done, but to prove that the IGF administered peripherally would arrive to the brain, a spinal tap was done before and after the treatment. And again, this is an old clinical trial, but the cerebral spinal fluid was maintained, is kept in the freezer, and eventually they allow us to test. And so we were able to show that even in this case, these are the patient before the treatment, not after the IGF. The IGF was not accessible to us, but the OTX2 is increased. So it gives us the idea that maybe it's worth it to explore. And so now what we are doing, we have done a couple of manipulation genetically where by doing some funky cross, MCP2 is still lacking in the mouse, but now we downregulate the expression of OTX2, either by injecting a virus with CRE in a mouse that is MCP2 mutant, but contain a FLOX gene for OTX2. When you put the virus, you will decrease the amount of OTX2 in the choroid plexus, like we see here in the bar orange, and that is sufficient to see rescue for the PV circuit, like here, the PV intensity and puncta is renormalized, and interesting, even the cortical thickness that is decreased in the mice lacking MCP2 is actually indeed maintained normal. The mice are better. In fact, if you look at their phenotypic score, a phenotypic score that was supposed to go up over time stays low. It's very different from a wild type that is 0. So it's not that we have cured completely Rett syndrome, but improve their health, the motor performance on rotor rod is better, and the survival is extended. And then another rescue that we have done, we use a FOXJ1 line that is specifically for epithelial cells of the choroid plexus, and again, cross with the OTX, so the CRE line for FOXJ1 with the OTX FLOX. The mice are still lacking MCP2. MCP2 is not there, and yet these mice do better. And the female also seems to be less symptomatic. And so, just to conclude, because I don't want to go too much over time, this is a glimpse of what we are doing. By starting from a very simple test like the optomotor task, we identify a phenotype that is maintained, abnormal phenotype that is maintained between animal to human, could become potentially a cortical biomarker for function in the disorders. On the basis of understanding what circuit underlies such disruption or participate to the disruption of such function, we were able to identify target for intervention and develop their targets. The last point I didn't present. And then last but not least is the thanks to everybody who did the work. Many of these people are Italians because it's easier to work with Italians with Americans. And my long-lasting collaboration, Takao Hensch, Chuck Nelson, Chin-Fei Chen, Graziella, Stefano Panzeri, Valeria, and the supporting agency. But the biggest thanks is to the Rett family, because they are the ones who really believe that basic research is needed to find a cure for their daughters. And this is Charlotte, this is Luca, my oldest son, and we work with the patient. We cannot cure them, but we can enjoy life in the pool with them. Thank you.
Beautiful lecture, which opened so many questions that I am sure that question will be asked from the audience. So we have 5 minutes of discussion.
So, Michela, especially your last results would suggest that are secreted factors that are mostly responsible for Rett phenotype. So, cell non-autonomous factors. Since the cells in the cortex are a mosaic, so you have some cells that have MACP2 and some that don't, are they any different in their morphology or physiology?
So it's an excellent question, of course, from you. I don't expect anything less. The one thing I would say, I would not say that the non-cell autonomous factors are the most controlling or inducing Rett syndrome. That is not correct, okay? Because we know if you remove MCP2 from every single cells, PV cells, pyramidal cells, any cells that you can think of, you will have almost any in the brain. If you do in the periphery, you remove everything from the periphery, non-brain-related periphery, you don't have Rett syndrome. But any cells in the brain will give you some degree, more or less, of Rett syndrome. And if you reintroduce in that particular cell, that cell feels better and the animals feels better. So there is definitely a cell autonomous factor. MCP2 is very important into the cells to determine their well-being and their cognitivity. However, they are also non-cell autonomous. And definitely, so we have not yet looked what happened for the cells that has or doesn't have MCP2, and when you do with this cell autonomous, as non-cell autonomous factor. We are doing now 2-photon imaging where we are able to see if the cell has or doesn't have MCP2 just from crossing with some ex-tomatoes that Pietro somehow knows all the crossing. And so we are able to see this cell has and this cell doesn't have. How do they respond to ketamine, for example? However, so we are doing that, or to OTX2. We don't have the answer yet, and yet also is quite difficult because the brain constantly changes. And so if you look at P15, or if you look at P30, if you look at P110, and everything in between, the things are changing. So it's like a moving target. So we will try to do as best as we can. The advantage of the females is that they have a slow progression. So the idea is to start at P30 when they are definitely asymptomatic, definitely should be okay, and see, is the one lacking already showing some defect, but because you are in an ocean of cells that they do have, your ocean of cells that don't have still can keep going, but eventually you tip the balance in favor of the lack of MCP2, but we don't know yet, but probably is in between.
Michela, do you have an idea? So you have shown that NMDA receptors on paralbumin neurons are critically important in all of this. So is there an idea what pathway, long-range pathway, would actually be involved in this? The second thing is that you have another thing, that you show that paralbumin neurons have a different kind of morphology, right? Mm-hmm. So they have more longer dendrites, more extensive dendrites, including more terminals. So there are two things, right? One is a feed-forward inhibition. Another one is a local process, which actually governs the inhibition to excitatory neurons, which are independent from one another. So there are two things at once, right? Which one is more important, which one the other? Do you have an idea of what actually drives the NMDA receptors?
Yeah, so the pathway for the NMDA receptor, we have sort of started to look at, however, are from homogeneity of the tissue. So you cannot be sure 100% that you're looking at the pathway from within the PV cells, okay? And so it's the AKT pathways that is misregulated very early on, and when we give ketamine, we do see a rescue. We cannot exclude that we are looking at the pathway activity within the PV cells. It's something that we would like to see, but it is an AKT pathway. Regarding feed-forward, feedback inhibition, and excited to inhibition. So most of our work was at the beginning, because this took a long time, very simple. PV cells onto pyramidal cells and a stop. But we know exactly what you said. PV to PV are interconnected, and they are responsible for the generation also of the gamma power. We are looking now. We have some mutant mice where you can remove the alpha-1 subunit specifically from the PV cells, and so we are trying to look in that way. There is a misregulation of the alpha-1 subunit we think in PV cells, so it could be that their circuit is also impacted. When we, for example, re-express MCP2 in every single cells, like the genetic manipulation that Adrian Bird has done, you do see that the PV to pyramidal cells is not rescue. That is set very early on, may take much longer. We may not have looked longer, but when you look at the gamma power and all the power, you see that there's some rearrangement, suggesting that maybe inhibition to inhibition tried to readjust, but we don't have yet. But we suspect that also that connectivity is affected. In addition, PV cells, there are two flavors at least. One is the large basket cells, the other are the chandelier. We haven't even touched that. Definitely they are innervating the axonal initial segment. What's happened there, that will have a significant impact as well. Also long-range inhibitory. And so one image that I show about the brain of the patient where you see that there is a spread of activation would suggest that a lateral inhibition is affected. And that would be inhibition to inhibition. And so that's where we are trying to focus now. So I think both are...
Can I ask a simple question? Probably neurons you are characterizing, or in which layer are they in?
So honestly, we look at every layer, okay? And we were curious to see if there was a layer specificity. We don't see, okay? However, we are now specifically looking at the thalamocortical preparation, thalamocortical. We know that the thalamic input arrives to the PV cells with a stronger input compared to the pyramidal, and that could be the starting of the disruption, because when we do voltage sensitive dye imaging, you see that there is a silencing when you stimulate white matter. Already at layer 4 there is a very much defect. Could be that they are the first to change, but with immunostaining, we don't see a layer specificity.
Michela, thank you. That was really interesting. I was intrigued by the last part because you talked about retina and choroid, and you are actually affecting the phenotype by interfering with layers that are by themselves normal, correct? Mm-hmm. So what is the link? What do you think is, how can you influence a phenotype at cortical level by manipulating the choroid? What is the end-up message?
So,
if you know it, or you can speculate.
We speculate. So OTX is a funky molecule, meaning that it's not produced in the cortex, as I said, the choroid and the retina, and travels. How does it travel? We don't know. Alain Prochance has shown over the years that is a protein that is able to jump from one cell to the other, because as this sequence that penetratin, that he has called penetratin, and create a penetratin peptide that allows to penetrate. Said that, okay, and seems that the activity either for the transfer or the uptake is necessary. So if you put the animal in darkness, you interfere with the production, but also to the movement of this OTX2. So the idea, and again, this is definitely a speculation, you have a constitutive production of OTX2. We do think indeed that MCP2 is regulating the expression of MCP2, because if you manipulate the expression of MCP2 in the choroid, you renormalize the expression of OTX2. So maybe they are linked together. And we know that the choroid is sensitive to activity. So there is a lab showing that the serotoninergic innervation of the choroid regulate their activity, and so maybe there is that induce. So there is somehow an overproduction of OTX2 that goes and bath the entire brain. So it's very aspecific, and in fact that's why I think we do see a general rescue, because we are not affecting just the visual cortex. There is maybe less OTX2, and so these cells that have this gain of function, even if they're still abnormal, they are better. But how it really happens, and what are the target of OTX2 once arrives into the PV cells? This is a work ongoing Takao is doing, and it's probably involving perineural nets and a lot. But so, sorry, not complete.
Thank you, Michela. I wanted to know whether you can intervene with OTX2 in order to rescue the rat phenotype only during the early period when you have this hypermaturation of PV cells, or whether you can act also late and have a
Yeah,
recovery.
So we have not done yet because we started with the genetic and the injection with the virus was done very early on, but that's exactly what we would like to know. Can we intervene later and still have a rescue? We know, not with the OTX2, but with the dark reading, when we were placing the animal in the darkness, even from P30, okay, and then that was specific for the visual system, was sufficient to have a better visual performance. So and because the things are, as I said, probably there is a maintenance role of this complexity from MCP2 in PV cells. The idea is that maybe even intervening later, we could some degree of renormalization is possible. We know for sure that MCP2 binds to the PV promoters, and when you play with MCP2, the expression of PV responds very quickly. But not necessarily, the connectivity takes time. So I would say that I think I will see some improvement given what birds have shown, okay? But I don't know if it will be sufficient. But that would be the hope, because then you can intervene at any age. Honestly, I think the later, the more difficult it's going to be. But because it will affect the entire brain, overall, you may have a better impact than just acting on one specific brain region or one particular cell.
So you're looking at the maturation of acuity in the first part of your talk. Are the paralbumin neurons affected before you actually can detect?
Yeah. Actually, so
how have you assessed that?
So the earliest recording that we have in the animal is P30. And at P30, the acuity is not dramatically impacted. The single unit is dramatically impacted. Okay? The acuity is maybe less, but not as different as is going to be later on. The PV cells are already abnormal at P15, but we don't have a recording that early, and that's a little bit more technical issue. However, using the female that again have this very long time course for onset, you can go very early, like P30 in a female is like equivalent of P15 in terms of phenotype. Already you do see some difference and some silencing, but it's not as dramatic as later on.
Thank you very much again. So the next speaker is Giorgio Carmignoto from the Institute of CNR in Neuroscience Institute of CNR in Padova. Giorgio came to work with us in the period in which the neurotrophin story began to take root, and he has been with us through all that period in which we collected some interesting rejections from for our early papers, which Giorgio called the gallery of horrors, and which then have been happily forgotten.
Se lo vuole mettere proprio incastrandolo così, che l'antenna così non perde il segnale. Le è rivenuta la possibilità?
So good morning to everyone. Thank you, first of all, for the invitation to this meeting in the memory of Luciano. And this is a picture of Luciano. I asked Nicoletta to send me some picture that I didn't have. And she sent me several pictures. And I choose this because, this is a time of relax in the old Institute of Neurophysiology. At the time when this institute was in Via San Zeno, at the time I was there, I was working with all these friends. And Luciano here is with friends, long life friends. And I like this picture because they are all smiling. As Nicoletta said before, I think smiling of Luciano is the best way to remember him. So it's a nice picture and it will be forever in my mind, this type of atmosphere that we all live at that time. So coming to science, the title that I choose for this presentation is this: astrocytes are at the center of the brain of neurons. Well, I think we all agree that the brain is made of neurons. Perhaps some of you disagree for the fact that the brain is also composed of astrocytes that can be so important to be at the center of brain function. I was convinced of that many years ago, but I think most of you are convinced that astrocytes are really important for brain function. And in the first part of this presentation, I will tell you something about the history of this, and then hopefully I will give you some recent data, unpublished data about the story of the ventral tegmental area and the role of astrocyte in reward mechanism. So how we did reach this conclusion? Well, this is a book from Ramon Cal that you probably know. I took two pictures from this book. And you can note, I have no pointer, but you can note the astrocytes and the pyramidal neurons there. And the process of the astrocytes are in contact with the soma and the dendrites of pyramidal neurons, but also the blood vessels, you see. So they make sort of contacts. And in some cases, they also embrace the soma of the pyramidal neurons. So there are a very stringent relationship between these two cell types in the brain. But at the time of Cal, we are at the end of the 90s, the idea that astrocytes can have a role in the brain was really, really unpredictable. But Cajal did many studies on neurons, but he also wrote a lot of aspects related to astrocytes. He was really a genius. And at that time, I put this sentence: the prejudice that the relation between neuroglia fibers and neuronal cells is similar to the relation between connective tissue and muscle or gland cells, that is, a passive or merely feeding supporting constitutes the main obstacle that the researcher needs to remove to get a rational concept about the activity of neuroglia. This, to me, is a suggestion that astrocytes and glia can be very active and not as simply supporting cells in the brain. This is our modern technique allowed to study astrocytes much better at that time. This is a patch clamp recording techniques from single astrocytes, and it's a simple movie to show you the fine processes of the astrocyte. You see, it's a zeta stack sectional, so you have these clouds of astrocytes. Very thin astrocyte can be really detectable by our modern microscopic techniques. So they are below the resolution, the optical resolution of a microscope. You can only see these clouds that of the fluorescence of the dye that were contained in the patch pipette. So the fluorescence is something blurry. It cannot really link this to a structure. And you can also notice that there is these nice processes that call end feet on the vessels. At the very end, toward the end, you see that counted the blood vessels. And at the EM level, you can see that the red label structures is the astrocyte processes that wrap completely the terminals, the presynaptic terminals and the postsynaptic part of a neuron. So they are in very, very intimate contact with neurons, and also with the blood vessel in the right part of the figure. So one single astrocyte can contact thousands of synapses. So this is simply something extraordinary. One single astrocyte can control a territory of more than 100,000 synapses. But what does it mean that they control? Well, the classical view of the astrocyte is that astrocytes provide trophic support to neurons. They can buffer the potassium, extracellular potassium. They can take up glutamate and GABA with transporter. And these are, of course, very important aspects in brain function. But this is only the beginning of the story, of course. And the paper that really was a breaking point in the physiology, in the story of astrocyte research, was this by Phil Hayden. It was published in Nature in 1994, and he he used a mixed astrocyte neuron culture, and he was able to show that when you stimulate a calcium increase in the astrocytes, by different means, he used bradykinin, mechanical stimulation, or photostimulation, as in this picture on the right part of the slide. He can stimulate a single astrocyte there, and then take a look at the neurons, and he detect that neurons respond with a calcium increase, which was mediated by activation of AMPA and NMDA receptors. So astrocytes release glutamate, which activate nearby neurons. This was a very, very important observation that was obtained in cultures, but it was something that illuminated immediately the interest and raised the interest of all the people around interested in glia, including me, that coming from the field of neurons. I was curious because I had some data on the astrocytes. So we performed at that time in the lab of Tullio Bolzan. We had the first microscope using UV light to detect calcium increase. So we can use at that time Indo-1, which is a ratiometric dye, very powerful. And in slices, we were able to show that when you stimulate shaft collaterals of the hippocampus and you monitor the astrocyte, you can see that the astrocyte respond with nice calcium increase that you can see going on different parts of the processes. And from time to time, this type of activity on the process is translated into a global response of the astrocyte that includes the soma and all the processes. So stimulating the release of glutamate from the shaft collaterals in the hippocampus, in the intact brain, let's say, in the intact brain tissue, could indeed evoke a calcium increase in the astrocytes. And we were able at that time to show that this calcium increase is translated into release of glutamate in the slices, which activate through glutamate receptors in neurons a calcium increase. So we demonstrated that this type of interaction was present not only in brains, in mixed astrocyte neuron culture, but also in the brain tissue. So it was a nice stuff. Also because led us then to reason on the contact of the blood of the astrocyte with the blood vessel. We thought, well, can this be a suggestion that astrocyte can work as a bridge between neurons and blood vessels? So they may be contributing to functional hyperemia, which is this neurovascular coupling mechanism that increases the blood flow locally when there is a local increase of activity. So this was an important stuff, and Michaela Zonta in the lab did this work, and The synthesis of the result that we have is this: when you have a neuronal activity very high, you increase calcium in the process of the astrocytes that are nearby the neuron, and these astrocytes have this intracellular calcium weight that lead to this calcium-dependent release of vasodilators that then act on muscle cells to relax the blood vessels. So this paper was, this study was to us a very nice period of research in the lab. So now the point, what was the point at that time? That we had to rediscuss the neurocentric view of brain function. And thus we came out with a proposal, we in the sense of the group of people that at that time was to some extent pioneering in studying this type of aspect. The concept of a tripartite synapse, which is, I think, absolutely nowadays demonstrated. That is, we had to think that the synapse is composed by presynaptic terminals, a postsynaptic neurons, and an astrocyte process. The astrocyte process can work both as a postsynaptic target and a presynaptic terminal, because indeed the release of glutamate and other transmitters typically used by neurons to communicate with each other are taken up, are recognized by astrocytes, because astrocytes express on their membrane the same type of receptors that are expressed at the postsynaptic membrane. So astrocyte can recognize the synaptic release of something, neurotransmitters, peptides, which trigger in the astrocyte calcium increase. This calcium increase can do a lot of stuff, and among these is the release of transmitters that we now call glial transmitters in this case, which can act both at pre- or postsynaptic sites in neurons. So they are contributing to synaptic function. And this review that Phil and me wrote in 2006, I underline the time of this, at that time we summarized the state of the art of the astrocytic research field in terms of the modulatory effect of astrocyte. And you see there are several aspects here. The release of glutamate can activate astrocyte, and in turn these astrocytes release glutamate, which can act presynaptically, acting on the mGlu receptors, modulating synaptic transmission, or the kinase receptor, which has also an effect on the probability release of transmitters, or they can act post-synaptically. They activate the NMDA receptors, the extrasynaptic NMDA receptors. I will tell you in a moment. They enhance synchronicity of the system. So if you have a release of glutamate, imagine from different sites of the astrocytes, they interact with different neurons and they enhance synchronicity of these activated neurons. Or they can also release ATP. So a lot of stuff, a lot of transmitters. Why? We don't know yet. But they release ATP, which can act both to the postsynaptic nearby neurons, acting on the ionotropic P2X receptors. But they can also act far away, because ATP is rapidly converted by endonucleotidase to adenosine. And adenosine is the most powerful inhibitor of presynaptic release. So from one hand, they respond to the local synaptic activity by releasing glutamate, activating nearby synapses, but also to far away synapses. They can control differently, possibly decreasing the probability of release, decreasing the activity. They use as a filter. There is an on-off filter. Here I increase the power of transmission, and there I decrease. So it's a sort of filtering effect. From that time of 2016, here are only some papers that follow. Papers that demonstrated long-time dependent potentiation of glutamatergic transmitters through the release of D-serine. So astrocyte release D-serine, coagonists of the NMDA receptor, and they can enhance, potentiate transmission through this mechanism. They also release glutamate following activation of the CB1 receptor. CB1 receptor is the most abundant metabotropic receptors in our brain. And it acts by modulating synaptic transmission when it's expressed in neurons, in neuronal membrane, but it also do different stuff, but equally important when it is activated in the astrocytes. There are different TRIP channels that can regulate the calcium increase, but there is a lot now of papers, and these are only those from, I think, up to 2010, something like that. The point is that I wrote this sentence from the Candel book that we all know, the neuron principle, what's the name, I don't remember, Principle of Neuroscience. And in the 4th edition of 2000, there was still this sentence: there is no evidence that glia are directly involved in electrical signaling. Signaling is the function of nerve cells. Okay? In 2000, after 10 years of data about astrocytes. I didn't take a look at the last edition of this book, but maybe this sentence is still there. I don't think so. But and in 2010, Nature, in occasion of the symposium, I don't remember exactly, but there was this commentary settling the great glia debate, because there are a lot of people who are still resistant to the idea that glial transmission has an important role in the brain. But this is not surprising, because the history of neuroscience was always shaped by struggle between different opinion, different vision. In this case, you may remember Camillo Golgi sustained the theory of reticularism, brain function idea. All the elements are connected electrically, while Ramon Cajal sustained the idea of a neuron, of neurons important, of the distinct elements in the brain. So, but maybe nowadays they are both right. I don't think that one of the two was wrong. And so nowadays, what we are proposing actually is that we can figure out that astrocytes are interacting with neurons in a different way. But the idea is that they do that in a with different features related specifically to time and space. So the time of the astrocytic action is much slower with respect to that of neurons. Neurons are fast. They have to transmit rapid information. Astrocytes modulate. And for modulation, you need time. To think, you need time. So to change your network, you need time. You need time to learn. You need time to adapt. And this is not a task of the neurons. This is a task of the astrocyte. No way. This is the task of the astrocyte. We propose 3 different aspects. And the first is the elementary calcium response. And indeed, we have plenty of evidence for that. Just a single synapse can be activated. It releases glutamate. It activates the tiny processes that are close by to that synapses. And in response to this activation, signal back to that synapses, modulate synaptic transmission there. Or you may have the B part, that this calcium increase can be transferred to another site of the astrocyte, to another processes, which is not really that tiny space. It can release glutamate or other factors in the distant part and affect distant synapses, as in the case that I told you of the endocannabinoid. This is a fantastic work by Alfonso Araker. He did spectacular works over the last 15 years. And in this case, you see, the neurons is releasing glutamate to activate the postsynaptic neurons, but the postsynaptic neurons is releasing endocannabinoids to depress the input. But the same endocannabinoids which act on CB1 receptor of that synapses which is activated, it activates the nearby astrocytes. And that astrocytes increase calcium because of the endocannabinoid release and release glutamate to the other synapse to to excite and to induce long-term potentiation. So you have, again, this filtering effect of a synapses which is releasing endocannabinoids to decrease the input, the glutamatergic input on themselves, but enhance the glutamatergic input of another synapses. So astrocytes can play around specifying the activity also in terms of the long-term changes of this type of synaptic circuits. Or you may have another aspect that is probably the less studied, that is the global response, because from time to time when you activate astrocytes increase the calcium all over the space, all over the processes. And this may mean something related to synchronicity of a network. Certainly there is a lot of major alteration in the tissue that is surrounding that astrocyte, so it's sort of what we call territorial synaptic modulation. And another aspect that I would like to tell you, I think is also interesting. The fact that glial transmission is granted by high affinity, slowly desensitizing receptor. This makes sense. We don't need, as I told you, we don't need speediness. Astrocytes are not as fast as neurons. Indeed, they need receptors that are not desensitizing, that are sensitive in a manner that they can keep the slowly increasing transmitter in the extracellular space. So they are involved practically only when you have motives to be involved. They are recruited to the circuit when the circuit needs to be modulated. And this means that they are involved when there is a lot of activity, which means memory, which means associative learning, which means a lot of aspects related to plasticity. I'm going too slow. I will not have time to tell you some data. I'm trying to go fast, if you don't mind. This is simply GFP, the glial acidic protein staining of an astrocyte. This is a use of a genetically encoded calcium indicator. This is simply to tell you that there is some part of the astrocyte, the very thin processes at the distal part of the processes that cannot be really detected structurally, but with the dye, you see these clouds. These clouds are the part of the astrocyte that are in close contact with the synapses. And what we can do is to express genetically, using this genetically encoded calcium indicator under a GFP promoter, only in cells in the astrocytes. This is an example of the astrocyte stain with this G-camp. You see this zeta stuck in the brain. And this is the resultant. You see the contact of the astrocyte with the blood vessel, blah, blah, blah. And here is to show you simply in vivo, in the awake animal. I put all this to movie. How huge is this activity of the astrocytes? This calcium signal is going up and down. These are making faster, but fundamentally the activity is there. Is a somatosensory cortex. And you see how big and huge and such a variety of signaling. And the point is, we have to understand the code. With Jimmy, we are trying now to set up and develop a software, a system which can analytically and precisely and accurately understand what is the meaning of all this stuff, but it's not an easy stuff. In the next 10 minutes, I have 10 minutes. Yes. In the next 10 minutes, I try to tell you something very new that we are doing in the lab. Actually, Marta Gomez-Gonzalo and Linda is a PhD student on the astrocyte control dopaminergic neurons in the ventral tegmental area. The ventral tegmental area is a very nice place to study. It's very deep in the midbrain. It's a very small structure, but very, very important. is close to the substantia nigra. So these structures are close by. These are dopaminergic structures. They are part of a very important mesocortical pathway, a mesolimbic pathway. The nigra and the nigrostriatal pathway is very, very important. Why is very, very important? Because I put this sentence from Bonci study to reinforce natural rewarding behavior and to attribute motivational silence to otherwise neutral stimuli. In other words, the VTA, together with the substantia nigra, but mainly the VTA, work as a hub system. The VTA receives input from a lot of structures, from all the sensory system, and then broadcast this information away. For what? To regulate reward, to regulate pleasure, to regulate aversion mechanism. So it's a very, very important stuff, very important nucleus. And at the basis of the nucleus is the dopaminergic cells. Is fundamentally a dopaminergic nucleus. 65–70% of neurons in the VTA are dopaminergics. They are also GABAergic, 20%, 50%, and glutamatergic neurons there. But the important stuff is the dopaminergic neuron. The dopaminergic neuron at rest, under resting condition, if you have an even animals, you just record, put a pipette on a VTA, you can find this tonic firing activity. But from time to time, you have a burst firing activity. So the switch between the tonic firing and the burst firing is the code of these cells. It's the code for aversion, pleasure, and reward mechanism. So the bursting activity is fundamental, informative, is how the dopaminergic cells work. And this is a paper in, I don't remember, this is a seminal paper, which show the reward prediction error mechanism. That is what? He put, I don't remember, is a monkey, is a mouse. He put in front of the possibility to have food, I don't remember. It was, I think, drops of juice. And it was recorded in vivo the dopaminergic neurons. You can see on the top that when the animal receives the juice, you have a discharge, an increased discharge of dopaminergic cells in a burst firing mode. So a very huge increase at the time, you see the reward here, of the drop of juice. Then they associated the drop of juice, but not really the drop of juice, before giving the drop of juice, will be present to you or just a clue. An auditory clue, a stimulus, a light. So they couple the light with the reward. So after 2 seconds from the light, the animal learns that it will happen. So when the light will come out, will turn on, when the animal was trained into that, the dopaminergic neurons increase firing, a burst firing mode at the time of the light, but not at the time of the juice, okay? What happens if then I will turn on the light and then I will not give the juice to the animal? The dopaminergic neurons are inhibited, strongly inhibited. So the dopaminergic neurons are coding the reward prediction mechanism. And this is important because this means that that is important to understand what is the controller of the dopaminergic neuron burst firing mode. And who control is mainly the glutamatergic input. So the glutamatergic input to the dopaminergic neurons is mainly the main pathway which controls the burst firing discharge. And these are a very simplified scheme of the control of the VTA of the dopaminergic control. We come from the lateral venula, lateral hypothalamus, the prefrontal cortex. Mainly these are the three main pathways that control the burst firing discharge in these neurons. So what we did is, we asked, do astrocytes respond to the burst firing? And what we did is to patch a dopaminergic cells, the green light, with a patch pipette containing neurobiotin. We distinguish dopaminergic cells because they have a huge IH current. So we are sure that these cells are dopaminergics. And we put also fluorophore, the calcium indicator, the red dye of the astrocytes are 101 to be sure that we are dealing with astrocytes. So you see this picture with the patch dopaminergic neurons, the astrocytes, and we will then induce a burst firing discharge of the dopaminergic cells and see what happened to the astrocytes. And what happened to the astrocytes is this. At the basal condition, there are no activity in general, but when you stimulate the burst firing discharge, you have an increased calcium in the astrocytes. And here is a movie of the basal condition of this. You can see more or less no activity. Then you have this. And here is the astrocytes responding. You see from one there up and down. They continue to be active. And this is the results. So when you switch from the tonic firing to the burst firing mode, you have a calcium increase in the astrocytes. The lady on the left is a female mouse because all these experiments have been done on female mice. If you done the same experiment on male mice, there is no response of the astrocytes. So there is no response of the astrocyte, absolutely. We also found that there is no response in the IP3 knockout mouse, of course, because IP3 signaling pathway is the main signaling pathway for the calcium increasing vascular. So we confirmed that these indeed are astrocytes, calcium dependent from IP3. So these are the data, the quantitative data. I'm trying to go fast. So what is known even in the VTA, that the dopaminergic cells, when they switch from the tonic to the burst firing mode, they release endocannabinoids. And since we know the astrocytes in other brain regions express CB1 receptors, then we hypothesize, of course, that the calcium increase, which follow the burst firing discharge in the dopaminergic neurons in the astrocyte, is due to activation of CB1 receptors. So we proposed, the idea was, well, when we have a switch on from tonic to burst firing, dopaminergic neuron release cannabinoids, which activate astrocyte, and astrocyte possibly release glutamate, which affect the other dopaminergic cells, enhancing the burst firing discharge. So the first part of this hypothesis is here. So astrocytes with immunogold in collaboration with Lorenzo Conti and Marcello Meloni in Ancona, and we were able to show that CB1 receptor is indeed expressed in the astrocytes. It's nicely expressed both in female and male mice. And that when you block the CB1 receptor with the EM251, you block the calcium increase in the astrocytes triggered by the burst firing discharge. So you block. What about dopamine? My goodness. The dopaminergic neurons, when they switch from tonic to burst firing, they release endocannabinoid, but they also release a lot of dopamine. So maybe dopamine has also a role. So we took a look at the dopamine receptors, and indeed astrocytes express D2, D3, and D4 dopamine receptors in the membrane. So maybe also dopamine is involved. So let's block the dopaminergic receptor and see what happened to the astrocytes. And it happened the same. If you block with etiqulopride, which is a D2-like antagonist, you block the calcium increase in the astrocytes. So the calcium response of astrocytes to the burst firing discharge is mediated by both dopamine and endocannabinoid release and co-activation of D1, of D2, and CB1 receptors. And And this is nice because Fiorenzo Conti and Marcello Vanone were able to show that the same astrocytes in the VTA express both D2 and CB1 receptors. And in some cases, these two receptors are close by. So possibly they are synergistically interacting to enhance the calcium increase. And indeed, you see, we block the burst firing induced calcium increase in the astrocytes. Just blocking one single pathway is enough. So this means that probably they need both. They need co-activation. Maybe there is also heterosynaptic receptor. So in conclusion, let's skip the conclusion, go, if I have 3 minutes, go very fast now. You have to be very, very fast. You also to be very fast to accompany me on that. So what we did is to perform the plasticity experiment. So we patch in the ventral tegmental area, not easy, the ventral tegmental area, 2 neurons, 2 dopaminergic neurons nearby. We induce the burst firing discharge in 1 neuron, which we know activate astrocyte. And then we record the glutamatergic input to the other neurons and see what happened to the glutamatergic input. The hypothesis is that the astrocyte activated by the burst firing discharge of neuron 1 release glutamate, release dopamine endocannabinoid, activate astrocyte, which in turn release glutamate, which activate the glutamatergic, which enhance the glutamatergic input to the other synapses. This is the results. You see, this is the basal activity. After 3 minutes, you have an increase. After 30 minutes, still increase. 45 minutes, still increase. So we have a long-term change of synaptic transmission there. But you know, it's present also not only in female. In male, you have only a transient increase. So this effect is consistent with the lack of calcium increase that we observe in male, of course. This is the quantification of a long-term change in synaptic transmission. And this is due to well, I have no time to go to that. Let's go to the last slide. And the last slide is this. What we were able to show is that when you have a switch from the tonic to the burst firing discharge in dopaminergic neurons are releasing 3 factors: dopamine, endocannabinoids, and nitric oxide. Dopamine and endocannabinoids induce a calcium increase in the astrocyte, long-lasting calcium oscillation in the astrocytes, which release glutamate. We checked on the presynaptic metabotropic glutamate receptor 1. If you block metabotropic receptor 1, you have no LTP. And these change the probability of release of the glutamatergic input, enhancing the release of glutamate, okay? But this change is long-lasting change only if there is the nitric oxide released by the dopaminergic neurons. If you block the nitric oxide, you have only a transient increase, but you have no long-term changes. So these are a complex situation in which you can detect a new form of plasticity of the input to the dopaminergic neurons, which is fundamentally important to regulate the burst firing discharge of these neurons. This is simply to say that as a conclusion that the brain should no longer be regarded as a neuronal network, but instead as an integrated network of interacting neurons and astrocytes. We are now also, this is a PhD project of Anna Maria Lia. We are looking at the start to understand Alzheimer and other diseases. And this is the important slide that recognizes the work of many people in the lab, but the work that I show you unpublished is made by Marta Gomez Gonzalo and Linda Requiem, is a PhD student. And also thanks my main collaborators and you for the attention. And I'm sorry for being so long and too long. And fast.
Okay, thank you very much, Giorgio. Okay, first question.
Thank you very much, Giorgio, for very insightful talk. I have a question on your general part. The fact that an astrocyte can independently control in a long-lasting manner several thousands or tens of thousands of different synapses poses the same problem that neurons face, and this was addressed in the past 15 years, about how a neuron controls long-lasting different synapses. So the same question now can be addressed for astrocytes. And in neurons, we know that there is a local translation of targeting of mRNAs, and local translation of mRNAs plays an important part in distributed and local control. So my question is, what do we know of local control of gene expression in astrocytes?
Almost nothing, of course.
So we should study it.
We are so ignorant about astrocytes still nowadays that we have obtained a lot of good evidence for an active role of astrocytes in synaptic transmission, but from a genetic point of view, all this stuff is really poor. A lot of work is done by Ben Barres on that, and he has done a lot of good studies on that. But you raise a point that, of course, is worth discussing, because it raises a question also of the plasticity, of the significance of the plasticity. And what I would say, but add to what in general I told you, is that we certainly are aware that there are also structural plasticity of the astrocytes. So there are data from Alfonso and others. They show that when you have an LTP established with the contribution of the astrocytes, then the processes of the astrocytes are moving away from this site. And so we don't know the real-time course of these events, but it's probably faster than what we think it can be. So you have also a sort of continuous interaction, but can be also structural. It can be also from regulation that we are not really aware, but they are occurring. Like microglia, this is another stuff of glia. Microglia are probably contributing to the plasticity in a different way, probably not through release of transmitters, but structured by unwrapping the synapses, by clearing the synapses. They can specify the synaptic transmission in a way that is important. But again, the point of the gene, of what is happening, that is something that needs to be addressed.
So 2 questions. One was, what do we know about the development of the astrocytes in terms of the development of the circuit, that part of contribution do they give? And the second question, you always talk about astrocytes and excitatory synapses. The network, there's the other elephant in the room, is the inhibition. How much impact do the astrocytes have on inhibitory transmission? And are we not missing some part, and that's why it becomes difficult to understand their contribution to the overall network activation?
Yes, there are many data about the role of astrocyte development, and specifically there are mechanisms that clearly has been revealed concerning the development of a synapse. There are the role of thrombospondin, so there are a lot of work, mainly by Ben Barres. They clearly show that.
And that's very early.
That?
Very early when the
that's true.
Form.
That's true. Well, you know, when you think of different timing, then during later development, this is something that needs to be associated with what we know about the role of astrocyte in plasticity. And you know, we were discussing even with Luciano at the time, because the story of the astrocyte will coincide with that period, more or less. And with the fact that we were thinking that astrocyte can release neurotrophins, and indeed, this is the case today. And we were thinking of the role of astrocyte in plasticity and visual cortex, and up to now, we have no real data about that. But I think that it's simply because we didn't take a look at the right place.
What about inhibition?
Pardon me?
Inhibition.
Yes. Well, this is a big story. Yes. Astrocytes, we showed recently that astrocytes are responding to both parvalbumin and somatostatin neuron signaling. They are responding to these two interneuron classes differently, because to parvalbumin they respond immediately but they depress. To somatostatin they respond immediately and they increase due to the somatostatin. So the response of astrocyte to somatostatin interneuron is due to both GABA and somatostatin, while to parvalbumin only to GABA, probably because there is such a tendency of the GABA receptors to the GABA B receptor to depress. Maybe there are some depression that is avoided by the somatostatin, but in any case, the point is, and this is well taken by you, are astrocytes involved also in the excitatory circuits? No, of course, no. And of course, they probably modulate inhibition in the same manner. There is, however, a paper by Alfonso in eLife last year, if I remember correctly. He was able to show that depending on the activity, on the input activity, and on the contribution of interneurons, astrocytes release either glutamate or ATP. Okay? So it may be the case that depending on the input, they release glutamate to excite, they release ATP, that is adenosine to depress. So there might be a sort of circuits. You know, astrocytes are simply, well, I would not to say that. This is something that I said many years ago. They are not simply, instead, adding a level of complexity in the system. They are accompanying neuronal activity, always, always. So when we think of what neurons are doing, we are also to ask ourselves, and astrocytes, are they contributing? Yes, and how? Maybe enhancing, maybe not, but they are contributing.
Okay, I have a very general and a bit provocative question. I mean, the fact that astrocytes contact the vessels, it means they contact basal lamina. And this is the typical hallmark of polarized cells, that they have a basal and apical compartment. And indeed, during development, they do have a basal and apical compartment. So radial glia has a basal and apical compartment. And astrocytes regulate neurovascular coupling, which is central in the regulation of brain physiology. So of these two aspects, modulation, so you may think that, I mean, what astrocytes do is to integrate, so they would be polarized cells in the sense that they integrate neuronal signals in order to signal to the vessel. So there would be unidirectional flow of information. So, you know, to what extent you think astrocytes are, you know, regulating synaptic plasticity, or this happens, but is relatively minor aspect with respect to the fact that they must have to integrate neuronal function in order to adjust the vasculature with the neuronal activity.
Well, they are multitasking. They are multitasking. Probably they are doing many things, and we have evidence that we are doing many things. They can distinguish. They are not simple target that respond in the same manner. No. They are synaptic circuit specific. They respond to that type of transmitter because they are in the circuits in which that transmitter will release. The astrocytes from, I would say, from the visual cortex probably do not respond to dopamine because there is no dopamine there. So there are no receptors there. So we have to think that astrocytes are not the astrocytes. There are many astrocytes. They are different. I didn't tell you, but in the VTA astrocytes are totally different from the other astrocytes. There is a paper by Bonci who studied structurally the astrocytes there. They are smaller, less processes, they are different. So we have to start thinking of astrocyte as a very large and complex type of classes of cells.
No, this is certainly true because there has been a very recent paper of single-cell RNA sequencing throughout the brain, and they clearly show that there are at least 5 or 6 different types of astrocytes that have very different molecular signatures. So that is certainly true.
Yeah.
I have a very simple. The experiment you showed in slice physiology were all done at very early ages, like P15 to P20.
Yes.
Is what you have told us a developmental phenomenon, or is it something which happens in mature brains as well?
I can anticipate you. These data are not published, but we are ready to, the paper is ready to send, but in a confidential manner, this situation. We repeat all the data in the adult. And if you keep this confidential, I would say that in the adult, the LTP, the astrocyte-mediated LTP is present also in male. Probably the CB1 and the D2 receptor are changing.
The males are slower than the females, which we all know instinctively, but I feel better.
Giorgio. Giorgio, is there any evident adaptive or even evolutionary advantage in the male to female difference? Or does it arise from other differences? Is that a consequence of other metabolic differences between males and female? Why are male astrocytes so different from? There must be some explanation.
The question is a big question, of course, but if I limit this to this system, to a VTA, the data that we have suggests that the defective LTP is due to a defective CB1 receptor expression, because I didn't make you notice that the CB1 receptor in the astrocyte from male is very low with respect to that in female. Okay. Since we have found that the LTP in male is established in the adult stage, probably there is more CB1 receptors. So probably it's simply a stuff of signaling, specific signaling for the part of a BTA. From the rest, I'll know.
So thank you for this mind-opening presentation. Maybe it's my ignorance, but do these filtering properties of astrocyte contribute to receptive field tuning or shaping?
My immediate intuitive, instinctive response is yes, why not? Yes, of course. But no one had taken a look at it. So I think it makes sense. Certainly, as I told you, as I mentioned before, astrocyte may work as a factor which, to some extent, enhance the specificity of that signal. Why? Because it may increase the efficacy of glutamatergic synaptic transmission when that glutamatergic synapse is highly activated. So in this sense, they increase this. But at the same time, they decrease the activity on nearby synapses through ATP adenosine. So it's an on-off mechanism that can be working.
I mean, what's interesting about this is that you are saying that glutamatergic synapses are mainly expressing mGluR receptors. Is that right? Did I get this right?
No, no, I was saying simply that one of the mechanisms by which astrocytes enhance glutamatergic synapses is through activation of presynaptic mGlu1.
Right. So if that's true, these synapses are mainly activated by specific pathways, not all of them.
Absolutely.
And the one which comes to mind is these from higher to lower areas, the so-called feedback or top-down. So in that sense, it's interesting that they are slower and presumably more integrative than the bottom-up, more specifically in tune with what has to be. So in that sense, I think your suspicion is probably not far off.
Yeah, I agree. But again, we have to say that the variety of the mechanism of the astrocyte-mediated plasticity are so high nowadays that it's surprising. It would be not a surprise to see many more in the future because we are really at the very beginning.
Okay, so now we will stop. And do you still have?
I was just saying that the bells of St. Stephen told us that it's time to go to lunch.
Okay, and we will come back here at 2:30.
So good afternoon. I'm told by my chairman that I can start. So, what I'll do in this moment is I'll try to start with. I'm a bit moved. So, in describing Luciano in a period in which many of you in Pisa did not have his presence, because Luciano spent from 1996 to 2005 at CISA in Trieste. And this is a picture courtesy of Nicola Origlia, and probably he gave it to me because he wanted to show himself. Anyway, this is Luciano with some of his collaborators, the early Nicola Origlia, Nicola Kuzniewski, and a senior postdoc in his lab. And this is the terrace of the so-called Bella Vista, that was a nice palazzina in close to CISA main building in Trieste. So Luciano, from 1996 to 2005, came to Trieste. He was, at the time we were setting up neurosciences at CISA. In 1996 already neurosciences were quite a big enterprise, but despite the fact that the labs, the neurosciences were well established at CISA, the logistic of the lab was quite complicated, and this is my personal history is a constant. And my lab was not at CISA main building, was in Area di Ricerca. It was a very nice and big lab, but because of Luciano, and because the electrophysiology labs were at Bella Vista, I decided to put an outstation of my lab at Bella Vista so that we could interact more closely. And two important postdocs in my lab, important because they have been very important in my group, were Simona Capsoni and Enrico Tongiorgi. They were based in Bella Vista, and we interacted very, very tightly with Luciano in those years. And those were very productive and exciting times, since both Luciano and myself, we were in some way exiliated, because we were singles from our families that were not in Trieste. This morning someone said that, Michela said that he was always the first in the lab, and this is absolutely true. And this was even more so when he was in Trieste. And also he was the latest in the evening, and that was a remarkable endurance that he had, but he never really suffered or complained for it. It was his passion. What I'd like to do is to switch slide, okay, and I would like to take you in the beginning through some of the work that Luciano did in those years, only to give you the flavor of some important interactions that we had. So those were, when I first met Luciano was first in Pisa, in what we call the heroic times of neurotrophins and visual cortical plasticity. There have been, and these are the papers that you all know. I was a passenger, friend, collaborator in that enterprise. And there is one paper that Luciano, following the paper by Nicoletta, in which antibodies were cell lines making antibodies were implanted in the ventricular, so in order to give continuous inhibition. There is this paper by Luciano in which I think that for the first time a treatment that would extend the critical period of visual cortical plasticity was described. Although what have been later called through the seminal work by Michela, Takao Hensch, all people in this audience about opening, reopening, removing breaks of plasticity and this. I like remembering that this type of treatment, I remember big discussions with Luciano about what might have been the mechanism around this. And of course, one prime candidate was cholinergic innervation. And so this is what Luciano in those years was very interested. This is a paper by Luciano and a PhD student of his, Emanuele Pesavento, in which they injected with a recording pipette in the visual cortex some new reagents that had been developed in the lab, a neutralizing monoclonal antibody to TrkA and a new form of a scavenger for NGF that was based on extracellular TrkA domain. And by this treatment, he was able to show that the developmental loss of long-term potentiation that is normally observed in the visual cortex could be rescued by inhibiting the NGF-TrkA interaction in situ locally, and he could show that the cholinergic system is involved in these NGF actions. And this is, in a way, a sort of you all know this work by Takao Hensch, in which links one cholinergic break limits plasticity in adult visual cortex, and it is possible, although it has never been tested or proven, that there might be a connection between these facts, and if there was a connection, this would vindicate Luciano's early interest in working on a neurotrophin in visual cortical plasticity that had lost interest because of other neurotrophins that for that type of experimental paradigm had acquired a greater interest. And so I like to mention that this was an important implication, I think, in some of Luciano's work in those years. A second aspect of Luciano's work in those years stemmed from the fact that in the lab we had just described that the RNA for BDNF is transported along dendrites in an activity-dependent manner. And so these were experiments in which you can, I should point it here probably. No, anyway. You can show that as a function of electrical activity, the mRNA of BDNF moves along the dendrite. It can move a very long distance. This is in cultured hippocampal neurons and also in vivo by doing treatments that affect So based on this, an activity started in which with Luciano, and you see here a number of papers that we published with Simona Capsoni and Enrico Tongiorgi. Basically, this the two labs had our own projects, but this was a very tight area of interaction. And what Luciano did was, with Simona and Enrico, was to study the activity-dependent targeting of BDNF mRNA in dendrites of visual cortical neurons as a function of activity, And there were many important findings that have been found here. One important fact is that there is a differential regulation of messenger RNA and of different isoforms. And we all know that this BDNF is a very complex RNA splicing form, and so I'll come back to this. And also the mismatch between BDNF mRNA and protein. So if you find BDNF mRNA, this does not necessarily mean that you have BDNF protein. In fact, it is rarely the case, and the translation is very strongly regulated, and the translation is also very strongly regulated in a isoform-dependent manner. And so these type of studies established an important concept in the field of visual cortex. This is shows that after dark rearing, the dendritic localization of BDNF mRNA in the visual cortex is lost. And in this paper by Luciano, this is a splicing pattern of BDNF mRNA, and you can study different isoforms. This is a double in situ hybridization, in which in orange or bluish you can have in situ hybridization for exon 4, that is the most activity dependent, or exon 3, and you can see either in blue or orange, or the reverse, that only exon 4 is dendritic, and in the same neuron, exon 3 is not. So this brought the final conclusion, what Luciano in his paper calls a spatial code for local BDNF actions, according to which in a pyramidal neuron, if you have BDNF mRNA here or here, it makes a big difference. And it makes a big difference to where, locally, where and when you make the BDNF protein. And this is under translational control, so this is a concept that now we are used to think in these terms. I think that when you see in a paper BDNF mRNA or BDNF Western blot, that's not the story that you need to see. You need to know where and when BDNF is expressed. And this comes from this work in which Luciano put all his insight and his passion together with Enrico Tongiorgi and Simona. So what I will do now is some work that I'll tell you one project that we are working on at the moment in the lab. It's a new project, is in recent years. I'm glad to see that Francesco Gobbo, who was the student who brought this project to success, who is now in Edinburgh, he came he can. So this work stems from what I was telling you about translational control of dendritic messenger RNAs. The idea is that we all know that if you want to characterize neurons that belong to a memory, these are called engrams. The idea is that memories are encoded by cell assemblies. However, well, this is work from Tonegawa's group and others in which they exploited a mouse that had been developed by Mark Mayford, in which you can genetically tag in an activity-dependent manner neurons based on their activity. And you can tag them with a reporter gene, that in this case is an excitatory channel rhodopsin. And you can do this in an inducible way so that you can activate it in a very well-defined time window that is linked to a behavioral task. And if you do this, what you can show is that by shining the light on and off, you can induce, in this case, a freezing behavior, a fear response in a mouse that had been conditioned that had learned the task. And so this is how you can, by genetically tagging neurons in response to a learning task, you can recollect the memory for that by reactivating those neurons. So this is the current trend in memory research. However, decades of work have taught us a paradigm that synaptic plasticity, in fact, is how we think that memories are encoded and brought about. And here are just some examples of correlations between treatments that affect LTP also affect memory and vice versa. However, the causality, cause and effect relationship between LTP and memory has not yet convincingly been proved. And there is also another problem, that the cellular engram paradigm posits that a cellular engram is a collection of neurons excited. The question is, this has a cell-wide spatial resolution. So the question is, there is a gap in scale there. Can we probe the engram concept at the synaptic spatial scale? Can we label spines that are potentiated in response to a given learning task, and can we map them and or think of reactivating them? So this is what you need new experimental tools to do this, and this is exactly what Francesco in his PhD thesis did. So the question that he addressed is, can we map potentiated synapses in living animals? Is there anything like a synaptic engram, a collection of synaptic sites that encode the memory? And can we reactivate potentiated synaptic inputs? And is this necessary and sufficient for memory recall? So the idea is to exploit the normal process whereby RNAs that are transcribed by de novo transcription following synaptic excitation are transcribed, they travel in the dendrite in a translationally repressed way. At synapses that have not been activated, these RNAs are not translated. At synapses that had been tagged by something that is called a synaptic tag that lasts a few hours, for the time of, sufficient for the time of this RNA to reach there. This tag allows the translation of the proteins that are necessary for potentiating that synapse. So we exploited this mechanism, and this is to express reporter proteins at potentiated synapses in cultured neurons and in vivo. So what we did was we did a construct whereby we take a reporter that in this case in the first case was channelrhodopsin, but it can be any reporter, and as I will say at the end, and we equipped it with two types of signaling: RNA targeting sequences that provide activation dependent dendritic localization and translational control, plus taken from a gene that is an inhibitory gene that is potently excited by synaptic activity, that is ARC. And also we put a protein tag that will target the locally translated protein into the postsynaptic density. So this is a bipartite protein targeting sequence that had been shown to target proteins into the postsynaptic density. And our control constructs are constructs that have the RNA but not the protein sequence, or protein that has only the protein sequence but not the RNA sequence. And to cut it short, what we can show here is that if you express the three constructs in neurons and you image them, then you see that only with the construct that has the SA, both the RNA and the synaptic targeting sequence, you have a very specific localization in the spine. And you can show that treatments that induce potentiation, such as BDNF or depolarization or chemical LTP of different sorts, only increase the synaptic labeling in those constructs that has the RNA and protein targeting sequence. You can show that, andare indietro come si fa? Così, sotto. So, you can show that these are potentiated synapses by various means. They are larger spines, which is a proxy for potentiation, but you can also use a reporter for exposed AMPA receptors, that is an AMPA receptor fused to a GFP protein that is pH dependent, so that when it is exposed, it becomes fluorescent, and this is shown here. This is a synapse that expresses AMPA receptor, and it also expresses our reporter. And the neighboring spines that do not express, so this is shown in a very high correlation, and if you use the construct that does not have the RNA targeting sequences, there is no correlation. So this shows that this probe is expressed at potentiated synapses. And you can then show that you can reactivate, you can do 2-photon glutamate uncaging experiments, and you can show that in the stimulated synapses, but not in the neighboring synapses, or not after without stimulation, you have a very high stimulation of potentiated synapses. This is shown here, in which you are stimulated this synapse, and 1 hour after you see that this expresses locally the channelrhodopsin protein. And if you measure the change in volume, that is a proxy for potentiation, and the expression of channelrhodopsin, you can see that you have a profile astrocyte that is very much reminiscent of a potentiation, of an LTP. And this is protein synthesis dependent, because if you do this in the presence of protein synthesis inhibitors, you have what would be an early LTP, and you don't have the expression of the reporter. So this is a probe that is expressed in a translation-dependent manner, in an excitation-dependent manner, at spines that have been potentiated. And you can also show that by shining blue light on the channel rhodopsin that is expressed in synapses, you can in neurons that also express a GCAMP calcium reporter, you have a very strong calcium influx. You have activation of calcium calmodulin kinase 2 here, but not in the dark. So you have a light-dependent activation of the spine, and you also can show that you have GFOS expression in the nucleus. So you can reactivate synapses in culture at the moment by shining light on the channel rhodopsin that is locally expressed in potentiated spines exclusively. What Francesco did, he also did the in utero incorporation to show it in vivo. And what he did was you express this probe in the hippocampus, and then he takes mice and he moves them from a home cage to a new context, and then he images ex vivo. And what you can see is you have a filler reporter that labels a neuron, but then in these you see these two fields are represented here and here, and the channel rhodopsin probe is only expressed at selected spines with no labeling of the soma. That is very, very important for our future experiment. And you can also see that the number of potentiated spines in the novel context is greatly increased, more so in CA1 region than in dental gyrus. This allows to map potentiated spines in vivo. And so what you can do is you can do some statistics on how a potentiated spine is located with respect to neighboring spines, and what you conclude is that you can show what is called synaptic clustering, that is, potentiated spines in the hippocampus are closer than chance to each other, so they construct real synaptic clusters. So, this is a platform now that allows to express reporter and actuator proteins at potentiated synapses. You can map potentiated synapses in vivo in areas engaged in memory. You could, in principle, reactivate previously active synapses, and you can also do something that we are doing currently. You can profile molecular differences in potentiated synapses. You can isolate with some suitable reporters the potentiated synapses out of the synapses that are not potentiated, and do some proteomic studies in this. So our ongoing work is the following. What Marco Mainardi, Ajesh Jacob, and Andrea Faraone in the lab are doing, they are repeating the memory recall experiment in a fear conditioning paradigm to ask the question, is the reactivation of potentiated spines sufficient for memory recall? The idea is to exploit mice in which this channel rhodopsin probe is expressed selectively at potentiated spines, as we showed we can do, and then reactivate them and see if shining the light in this context after fear conditioning in this context is sufficient to reactivate the memory. If you express a protein, a reporter probe that is a proteomic handle, you can couple this with mass spectrometry. This we are doing in collaboration with Alessandro Ori in Jena and Alessandro Cellerino here in Pisa. And we can do mass spectrometry. We are collecting the first available samples of proteomic data from potentiated spines, trying to see what is the molecular signature of a potentiated spine. And what we are also doing is, the discussion this morning was about astrocytes, and of course there is an interest if we can construct a similar probe to express locally activated portions of an astrocyte, and also to map potentiated inhibitory synapses. To conclude, this is the group that here in Pisa is working on this. Francesco Gobbo, who is now in Edinburgh, Marco Ajesh, and Andrea Faraone. and our collaborators. The work was done in collaboration with Laura Canceda, who did for us, and Bruno Pinto, who is a PhD student from Scuola Normale in Laura's lab. They did the in utero electroporation. And I forgot here Tommaso Fellin and his postdocs in Genova. I would like to conclude by this. This is a trattoria Città di Londra in Trieste, and this is where Luciano liked very much to go for dinner. We usually left Bella Vista at after past dinner time, and this was his favorite place, and we had countless dinners and brainstormings with Luciano, talking of science, talking of families. We had young children, and this is the inside of this trattoria, so this is why it was called the Città di Londra. And it is a very strong memory for me to think of he was very relaxed there, and he enjoyed it. And so I would like, okay, no, this is important. Matthew Diamond, who was, when Luciano came at Sissa, he could, our biophysics sector was already established, and the natural place where he would come was in our sector. Daniele Amati, the director, who had this vision of creating neurosciences in a place that was only of physics and mathematics, had started filling the gap, and he had created a cognitive science department sector. And so he convinced, he called Tim Challis, who is a neuropsychologist from London, to come to Pisa, and Tim Challis recruited Matthew Diamond. And Luciano was convinced to go in that sector so he could strengthen a bit that sector. So although we belonged to different sectors, he was. And so I asked Matthew Diamond about his memories of Luciano, and he sent me this sentence that I would like to read with you, because Matthew Diamond, I can say, was not a particular friend of Luciano. He was a colleague. So this is a statement by a colleague, not by a friend. And he said, so in fact he said, there is no specific memory that I can tell you, but rather an overall impression that was reinforced countless times in various ways, that he was always modest, perhaps too modest in an era where it is common to be something of a self-salesman, extraordinarily serious, reliable and careful as a scientist. He was also the most transparent person I have known in the academic world, in that he never had a hidden agenda. He explained himself in a clear way, and whoever listened knew that that was the entire story. And I think this, from a colleague and not from a friend, is a very important statement. And so let me conclude by thanking Luciano for his example, his contagious enthusiasm, endurance, unrestrained passion, and ethical approach to science, academic matters, and to life.
Or otherwise we will move to the memories part. This is a little bit of a transition.
How do you know that a synapse is potentiated? A particular spine which you're looking at.
Yes.
How do you know that this is it?
So our catalog of proofs is the following. I went rapidly through that. One is that spine volume or increase always correlates with the expression of the reporter, and as you know, so-called single spine LTP experiments by Kasai groups and others establish an equivalence using increase in volume as a proxy for potentiation. So this comes from single spine experiments, and in our hands, spine volume always correlates with the expression of the reporter. Second evidence is the spines that express the channel rhodopsin or the reporter always also express exposed AMPA receptors, and that is with the pH-sensitive AMPA receptor expression. Third evidence is that you can do, I showed it but it was too fast, immunofluorescence with anti-phosphorylated calcium CAM kinase 2, that is known to be activated by an NMDA receptor, and this is also correlates in a very high correlation with the expression of the reporter. You can also do pharmacology experiments in which treatments that abolish the former also will interfere with this potentiation. So in vitro, we are pretty convinced that we are labeling not activated spines, but potentiated spines. And that is an important distinction. We are not mapping simply, I would say, activated spines as you would do with the GCAMP, but we saw on the converse, this is another evidence, we show that if you, GCAMP expression also will correlate with this, but that is activated spines. Here there is potentiated spines. There is a gap. In vivo, we assume that it will be so, but maybe, Francesco, you will say something. Va bene.
So, if there are no other questions, we will start with, we will do a number of short memories of Luciano, and the language is open, so you can talk English if you want, o chi vuole può anche parlare in italiano. And so the first person I would call is Ferdinando Sartucci.
Buonasera a tutti, grazie per avermi invitato a partecipare a questo evento. Scusate, mi sono fatto alcune annotazioni perché non vorrei che mi tradissero le mie facoltà in questo momento. Io ho iniziato a conoscere Luciano Domenici nel 1980. Era l'anno in cui preparava la propria tesi di laurea in medicina e chirurgia, che fece una tesi sulla prosopagnosia. E si laureò poi ad ottobre dello stesso anno con lode. Quindi decise di intraprendere la carriera clinica di neurologo e si iscrisse alla scuola di specializzazione e si è specializzato in neurologia il 15 di novembre del 1984 e discusse una tesi sul ruolo della dopamina nel sistema visivo dell'uomo. Ed è stato durante questo periodo che è nata, diciamo, la nostra amicizia, perché durante la specializzazione in neurologia lui ha iniziato a frequentare la neurofisiopatologia in cui io lavoravo, ero un dipendente, e quindi abbiamo iniziato un po' a lavorare insieme, abbiamo prodotto anche tutti questi anni diversi lavori scientifici, si era allontanato un po' dalla neuropsicologia e si era più affascinato dalla neurofisiologia. Abbiamo fatto, credo, 12 lavori insieme. Diciamo che questo portava anche un impegno clinico, Luciano non si è mai tirato indietro, neanche per le attività assistenziali o per quelle di tipo organizzativo che richiedevano, diciamo, che risultavano pesanti, diciamo così, per le sue condizioni fisiche, se me lo lasciate passare. E quindi siamo iniziati a frequentare, poi è andato, lui ha avuto un dottorato di ricerca nelle discipline neurologiche, è diventato ricercatore del CNR, ma abbiamo continuato, diciamo così, a collaborare, a stare insieme e a fare progetti anche finanziati, eccetera, per cui abbiamo continuato a frequentarci anche al di fuori dell'ambiente di lavoro. Poi si ricorda negli USA, io quando prima di andare in America sono stato ospitato prima di andare in America a imparare un po' di neurofisiologia della visione, sono stato ospitato nel suo laboratorio, poi gli ho sempre fornito collaboratori perché avevo un corso di laurea in tecniche di neurofisiopatologia e quando lui aveva bisogno di personale mi diceva se aveva qualche giovane valido, se glielo insomma, se glielo inviavo, sceglievo, glielo indirizzavo, diciamo, per i compiti di ricerca. E ne ha usufruito di molti. Qui vorrei ricordare soltanto l'Andrea Filippi, che è stata la prima, poi il Barloscio, l'Elisa Cerri, non so se c'è, qui non c'è oggi. Quindi. E poi, mi scuso se me ne sono dimenticato qualcuno, poi è diventato professore, lui è andato all'Aquila. Però tutto questo tempo ci siamo frequentati, abbiamo continuato a sviluppare dei progetti di collaborazione, dei paper, abbiamo scritto insieme paper, qualcuno è rimasto ancora nel cassetto, se vogliamo dire. Abbiamo condiviso anche degli aspetti, se lasciate parlare la parola, conviviali o di tipo turistico, quando siamo stati a giro, come succede per tutti. E ho condiviso purtroppo anche come medico i suoi periodi di sofferenza. I suoi inciampi e le sue condizioni di malattia che lo hanno portato spesso a ricovero in ospedale, spesso a dover ricorrere, diciamo, al mio intervento, che però non l'hanno, diciamo, un po' minato nel suo nelle sue condizioni fisiche, però non l'hanno mai minato nella sua mente e nel suo spirito, questo ve lo posso dire, perché nonostante tutto questo aveva sempre delle parole di incoraggiamento anche nei miei confronti, che anch'io ho dovuto condividere alcuni anni di problemi sanitari piuttosto gravi in famiglia. E lui, quando era ricoverato da una parte, io andavo da un reparto all'altro per mia moglie, a trovare lui e Mariolina, e quindi aveva sempre delle parole di incoraggiamento, diciamo, per me e per la mia famiglia, e che capiva che stava attraversando un periodo che pensava fosse forse peggio dei suoi problematiche. E quindi sembrava che queste condizioni fossero insuperabili, ma lui pensava sempre, aveva sempre qualche parola di incoraggiamento e di andare avanti, qualche parola di speranza. Ma ora vorrei passare a parlare di Luciano come studioso, perché secondo me tratteggiava la figura dello scienziato. Era una persona molto aperta, meticolosa, rigorosa nella procedura sperimentale, e tutto ciò emerge dalla sua attività scientifica, come abbiamo sentito in precedenza brillantemente anche da Antonino Cattaneo. Quando in genere ti lascia un amico più che un collega, un amico che fosse stato accanto per tanti anni, c'è un dolore come quello di perdere un familiare, un fratello, una sorella. È quella persona che si chiama e si interpella nei momenti di difficoltà, ma anche in quelli di gioia. E vorrei che tutti sapessero cosa faceva, come si comportava, ma soprattutto gli aneddoti più divertenti, dolci, quelli che lo hanno fatto apprezzare e hanno fatto capire quanto fosse importante per me il rapporto personale con lui e con la sua famiglia e tutti quei bei momenti che abbiamo vissuto insieme. Luciano sprigionava un entusiasmo e uno spirito vitale che continua ancora a rivivere nella mia mente e sostiene sempre il suo ricordo, rallegrando a volte anche la mia vita di fatica. Pertanto la sua morte, la sua scomparsa inattesa, Apertura mi lascia un grave vuoto, ma anche lascia una traccia luminosa delle sue virtù, perché la fede è stata la sua guida, quindi testimonia anche il libro Il Vangelo dei poveri. L'onestà era il suo ideale, il lavoro era la sua vita, la famiglia il suo affetto, i colleghi la realtà del suo universo. Ed in questo universo, che ci sono tante stelle e che sono sempre più rari, perdonatemi questa affermazione, c'è anche la sua, che secondo me è una fra le più luminose. E guardandola penso che tutti noi troveremo sempre conforto, sostegno, ma anche affetto. Quindi grazie Luciano per l'insegnamento che ci ha dato, per quanto ci ha fatto comprendere, intravedere, per quanto ci ha lasciato in eredità. E penso che continuerai a vivere ogni volta che io leggerò un tuo lavoro o un tuo libro.
Okay, so next one is Nicoletta. I'm trying to keep a little bit a chronological order.
Ok. Allora, anche io mi sono scritta qualcosa. Io ho lavorato con Luciano per circa 10 anni, continuativamente, a partire circa dall'86. Le mie memorie cominciano con Luciano che arriva la mattina e si inizia a lavorare sempre con un sorriso, con un atteggiamento ottimista. Il primo lavoro che abbiamo fatto insieme è stato nel gatto, l'elettroretinogramma, e usavamo l'aloperidolo, un farmaco dopaminergico. E mi ricordo Luciano che mentre lavoravamo faceva: beh, però se questo qui si mettesse nella canala del condizionamento e magari si vivrebbe meglio. E io ci ho pensato spesso nelle riunioni universitarie a Firenze. Beh, certo, effettivamente se ci potessi arrivare non sarebbe una cattiva idea. Nell'87 abbiamo iniziato a lavorare nel ratto. Era una bestiolina su cui non avevamo mai lavorato, quindi passare di specie poneva problemi, e quindi ricordo queste giornate passate semplicemente a rispondere a questa fondamentale domanda: o dov'è la corteccia visiva del ratto? O dov'è la fovea del ratto? Non c'è la fovea. Quindi io ho un ricordo di giorni e giorni passati con Luciano semplicemente a metter su un qualcosa che adesso può sembrare banalissimo, far venire fuori un elettroretinogramma da un ratto. Quando abbiamo cominciato poi a lavorare nella corteccia visiva del ratto, i problemi ovviamente sono aumentati, e qui però abbiamo dato, secondo me, un contributo fondamentale alla scienza, che purtroppo non è più usato perché è stato, diciamo, superato dagli eventi, che è l'eccentrimetro. Immagino che voi giovani non l'avete mai visto. Ok, allora, per misurare l'eccentricità dei campi recettivi delle cellule corticali visive che si proiettavano su uno schermo, per evitare di fare calcoli che al buio venivano anche male, avevamo prodotto una riga di plastica rivestita di un foglio con tante righine che avevano, diciamo, come scala l'arco tangente, e che davano immediatamente l'eccentricità in gradi del campo recettivo. E l'eccentrimetro è una parola di Luciano che veniva usata all'epoca quotidianamente. E l'altra cosa che ho sempre in mente di Luciano quando si registrava dalla corteccia visiva era: vai, cambia le mutande all'elettrodo, perché bisognava schermarlo, se no c'era ovviamente rumore di fondo e lo schermo era di stagnola, che veniva messa accuratamente, adesso si fa meglio ovviamente, e quello era affettuosamente chiamato le mutande dell'elettrodo, quindi se non funzionava qualcosa, prima o poi Luciano diceva: via, cambia le mutande all'elettrodo. Un lavoro in particolare che ho in mente di Luciano è stato quello in cui abbiamo iniziato a usare i potenziali evocati nel ratto che non venivano. E io mi ricordo Lamberto che ci sfotteva giorno dopo giorno: non siete capaci nemmeno di tirar fuori un potenziale evocato da flash, manco da reticolo. E effettivamente era un pochino frustrante. Poi una sera, tornando a casa, con Luciano si diceva: ma magari è perché non usiamo delle condizioni ottimali. In fondo il ratto è un animale notturno e non gli si dilatava la pupilla come si faceva nel gatto. Il giorno dopo abbiamo registrato senza dilatare la pupilla, sono venuti fuori dei potenziali evocati 100 millivolte. Allora siamo andati di corsa da Lamberto: Lamberto, abbiamo trovato il problema. Ma figuriamoci se era questo. Per cui penso nell'unica volta nella nostra vita abbiamo potuto dimostrare che aveva torto, perché c'era il potenziale evocato, gli abbiamo dilatato la pupilla, il potenziale evocato è scomparso. Forse avete ragione. A distanza di anni Luciano mi faceva: forse una volta nella vita si è avuto ragione. E un'altra cosa che di quel lavoro mi è rimasta impressa è che il programma che usavamo per registrare i VEP l'aveva fatto mio marito. E Luciano ogni tanto, stamattina cercavo di dirvi che Luciano faceva dei giochi di parole e delle battute fulminanti. E ogni tanto, quando veniva bene l'esperimento, lo sentivo canticchiare. Mio marito si chiama Tinivella di cognome: viva viva il Tinivella, guarda l'onda com'è bella. E quindi anche questo è un qualcosa che è legato alle mie memorie di lavorare con Luciano. Quando noi abbiamo iniziato a lavorare, abbiamo lavorato per tanti anni come gruppo, con Michela giovane, con Tommaso giovane, con Luciano, e prima ancora quando ero giovane io e lavoravo con Silvia, quando si dice che eravamo una famiglia, non si dice così per dire. Il lavoro era vita, ma non nel senso che escludevamo. Noi abbiamo importato nel nostro gruppo di lavoro, io ho importato mio marito e Luciano ha importato Mariolina, che ci vedevamo, però la giornata lunga di San Zeno era una giornata in cui noi si condivideva tutto e veramente siamo stati famiglia, e come diceva Fernando prima, poi è proprio un pezzo che se ne va, non è un collega, e non è nemmeno un amico, è un pezzo di vita, è un pezzo di famiglia. Allora, le ultime due cose che vi dico, tre, via. Uno è il lavoro che Antonino rammentava, quello con gli ibridomi che producevano gli anticorpi contro l'NGF, che per me è un esempio di come semplicemente essere amici con persone di valore poi produce. Tornando in treno da Napoli, dove allora insegnavamo entrambi, Antonino tirò fuori dallo zaino un ciancicatissimo lavoro e fa: guarda qui. E questo era un lavoro sulla rigenerazione di Schwab, in cui lui usava anticorpi contro NGF. Quindi dice: questo lo possiamo fare nella corteccia visiva domani. Allora io sono arrivata a casa alle 10 di sera e ho chiamato Luciano. Ho detto: Luciano, domani. E ora, domani forse no, ma insomma credo 3 giorni dopo eravamo al lavoro. E così effettivamente è stato, come diceva Antonino, il primo lavoro in cui, manipolando molecolarmente in maniera anche piuttosto grossolana, se vogliamo, una molecola, si rallentava lo sviluppo e si influenzava il periodo critico. E le altre due cose riguardano due viaggi. Uno è stato un viaggio a Cambridge in macchina, in cui per risparmiare siamo andati tutti con la mia macchina. Quindi siamo partiti da Pisa, io, Carmela e Lucia. A Boulogne abbiamo raccattato Luciano. E qui, diciamo, da poveri provinciali, non ci è venuto in mente che Boulogne potesse avere 2 stazioni. Quindi l'appuntamento era alla stazione di Boulogne. Quindi io e Lucia siamo arrivati, Luciano non c'era. Abbiamo fatto fare un annuncio all'interfono, le professeur, Luciano, Domenici. Poi abbiamo chiesto, ma quale stazione di Boulogne? E siamo andati all'alta, dove Luciano ormai si era visto perso e ci ha visto arrivare. Dopodiché abbiamo varcato la Manica con la mia macchina, ma prima abbiamo dormito a Boulogne per risparmiare tutti e 4 in una stanza. E prima siamo andati a cena in un posto terribile, dove Luciano, per tirarci su di morale, continuava a chiamare l'oste Papa Fernand, e a distanza di anni faceva: ma com'era buona la cena di Papa Fernand, che abbiamo messo 3 giorni a digerire la zuppa di cipolle di Papa Fernand. Però Luciano, tutto contento e tutto allegro, la mattina dopo siamo sbarcati in Inghilterra con una faccia talmente tremenda che una signora ci ha guardato e ha detto: oh poverini, avete bisogno di un caffè, vero? E Luciano fa: sì, grazie signora. E l'ultima cosa è il primo viaggio in cui siamo andati fuori al Neuroscience, era a Toronto. Ma non era un caso che io sono stata mandata a Toronto, perché quell'anno era morta la mia mamma e Luciano e Lamberto hanno deciso che dovevo essere mandata fuori a prendere un po' di aria. E Luciano è stato il mio angelo custode, come lo era stato per tutto il periodo della malattia della mia mamma, nel quale ha cercato di incoraggiarmi, di aiutarmi a capire cosa succedeva, di non lasciarmi travolgere in maniera completa. E quindi ho questo ricordo di questo viaggio a Toronto, in cui non mi ricordo assolutamente niente di quel neuroscience, però mi ricordo di queste camminate con Luciano, in cui piano piano ricominciavo a parlare di lavoro e non soltanto di analisi che erano andate male o di peggioramenti, e che Luciano pazientemente aveva sopportato e aveva supportato per più di 2 anni e mezzo. Quindi di quel viaggio lì ho il ricordo di questo viso sorridente che mi aiutava a guardare di nuovo a un futuro. Quindi ho molto da dire grazie a Luciano. Non è mai riuscito a insegnarmi a fare della buona chirurgia, ma questo ha fallito anche Silvia, e quindi evidentemente deve essere una cosa genetica. Però è riuscito a insegnarmi a fare il punto chirurgico. Ecco, questo Luciano, con grande sorpresa di tutti, era riuscito a insegnarmelo e lo commentava dicendo: vedi, picchi e mena anche la Berarda è riuscita a imparare a fare il punto chirurgico. Come dire, mai gettare la spugna. Ognuno ha delle potenzialità che sufficientemente coltivate poi alla fine sbocciano. E quando penso alle lunghe chiacchierate che abbiamo fatto appunto di lavoro, ma anche di progetti di vita, di figli, di politica, ho sempre in mente questo sorriso e associo a Luciano una sensazione di calore e di forza che penso è rimasta in tutti quelli che l'hanno conosciuto.
Okay, now I have a short message from Yuri Bozzi, who could not. Yes. No, I read this briefly and then I can pass it over. So this is a short message from Yuri Bozzi, and because me, Michela, and Yuri, we all entered in Via San Zeno in 1989, which looks like ages ago, and so. This is what Yuri says. Dear friends, I'm very sorry not to be here with you to remember Luciano, as he would have liked, with a great day dedicated to science. Luciano has been for me a great example of dedication and passion for science. I like to remember him many years ago, it was 1990 or 1991, in the molecular biology lab of La Fontina, working hard to learn how to extract RNA from the brain. His hand was shaking, but he never gave up. Those were the years of my thesis, when I was desperately seeking, together with many of you, to study neurotrophin expression at the molecular basis of visual plasticity. Then, for all year I spent at CNR as a researcher, Luciano has been a guide for me. His door was always open to discuss about science and many other things. Thank you, Luciano. Now I give it to Professor Piccini, who collaborated with Luciano on aspects of psychiatry.
Thank you. Buonasera, scusate, io siedo un attimo perché altrimenti ho paura di dire delle cose diverse di quelle che poi penso, allora le leggo. Allora, buongiorno a tutti e grazie agli organizzatori per avermi invitato in questa giornata in cui alcuni di coloro che hanno conosciuto e lavorato con Luciano lo rievocano e lo ricordano. Ho conosciuto Luciano nel 1978, eravamo entrambi studenti interni della clinica neurologica diretta a quell'epoca dal professore Alberto Muratorio. Familiarizzammo subito per una simpatia che si instaurò reciprocamente tra noi. In quegli anni lui lavorava con il professor Muriore nel laboratorio di neuropsicologia con il professor Arena. Ci laureammo nello stesso giorno, alla fine di ottobre del 1980. Dopo la specializzazione in neurologia imboccammo strade diverse. Lui continuò a coltivare il suo interesse e la sua passione per la neurofisiologia, io iniziai il mio percorso verso la psichiatria. Dopo circa 20 anni, alla fine degli anni 90, ci siamo nuovamente incontrati. Fu un incontro molto bello, ci raccontammo quello che avevamo fatto in tanti anni e lui mi parlò subito di un argomento che disse: vedrai Armando, questa cosa ti appassionerà moltissimo. Mi parlò dei suoi esperimenti, dei fattori neurotrofici, dei concetti della neuroplasticità. Fui affascinato dai suoi discorsi. Passammo lunghi pomeriggi a discorrere su quell'argomento, su come trovavamo punti di contatto tra la sua prospettiva di neurofisiologo e la mia esperienza di psichiatra. Nacque così un sodalizio che è durato per tanti anni e che modificò profondamente la mia modalità di vedere le malattie mentali. Iniziamo a progettare protocolli di ricerca sui pazienti psichiatrici che attingevamo dalla clinica psichiatrica di Pisa, dove io allora lavoravo. Portammo a termine tanti lavori che ci dettero conferme e soddisfazioni. Furono degli anni di lavoro molto intenso, ma soprattutto di entusiasmo, di scambi di conoscenze e di progetti. Ci incontravamo nel suo studio dell'Istituto di Neurofisiologia del CNR a San Cataldo. Luciano era nei nostri dialoghi, pur nel suo rigore scientifico, sempre pacato, composto, delicato. Non amava i toni forti, anche se i concetti che esprimeva erano forti e vigorosi. Non diventava mai nelle nostre discussioni categorico assertivo. Le sue parole lasciavano sempre uno spiraglio di riflessione e rielaborazione, di verifica ulteriore. Tante volte stemperava il mio temperamento un po' più irruento del suo con poche parole che mi rimettevano in un canale di pensiero, che era quello che lui riteneva più giusto seguire. Devo confessare che amavo questo gioco che c'era tra di noi, in cui io svolgevo il ruolo del bimbo discolo e Luciano il padre saggio, che con poche parole, pronunciate con voce grave e pacata, risistemava le cose e le metteva al loro posto. L'argomento che trattavamo era poco conosciuto alla maggior parte dei colleghi che si occupavano di clinica in quel tempo. Decidemmo così di organizzare un congresso sui fattori neurotrofici e sulla neuroplasticità. Quelli della preparazione del congresso furono mesi di lavoro febbrile. Riuscimmo ad invitare molti studiosi internazionali ed il congresso che tenemmo nella sala convegni del CNR fu un successo. La mattina dell'apertura dei lavori ci trovammo davanti un'aula di quasi 300 posti, gremita, con tanti colleghi che non trovarono posto e restarono in piedi per ore per ascoltare i relatori. Luciano era raggiante, era il coronamento di uno dei suoi sogni, che era diventato anche il mio. Diffondere la conoscenza di argomenti che in quel periodo erano poco o per niente conosciuti ai clinici e che erano per certi versi una rivoluzione nel modo di funzionare del cervello e dei meccanismi che erano sottesi nelle malattie mentali. Il ricordo di quegli anni è un pezzo della storia della vita di Luciano e della mia. Fummo invitati in tanti posti a presentare l'argomento, anche alcune industrie farmaceutiche si interessarono a noi e ai nostri studi. Ricordo una frase di Luciano all'uscito dei laboratori di ricerca Glaxo di Verona. Disse: mi sono sembrati molto interessati alle cose che gli abbiamo detto. Abbiamo risposto alle loro domande in maniera efficace. Siamo apparsi per quello che siamo diventati con la nostra esperienza, due splendidi cinquantenni. Questa frase mi è rimasta impressa. Luciano era una persona rigorosa, non era avvezzo all'autocompiacimento. L'evento era stato per lui però così positivo che aveva aperto in lui un piccolo spiraglio da cui quell'emozione era riuscita a manifestarsi e a sfuggire. Ho nei confronti di Luciano tanti sentimenti diversi, di affetto per la sua umanità, per il suo senso dell'equilibrio e della misura, di ammirazione per la sua mente arguta e brillante, di rimpianto dei nostri anni verdi, di riconoscenza per tutto quello che ho imparato da lui. È facile parlare di storie, di fatti, di aneddoti, è sempre molto più difficile parlare di emozioni, di passioni, di affetti, di suggestioni. È l'eterna lotta tra la nostra piccola e fragile quota di razionalità e la grande e potente porzione di emotività che indirizza e regola il nostro comportamento. Queste poche parole che ho scritto di getto è avvenuto proprio così, fatti, storie ed un poco di emozioni. Grazie ancora una volta a Luciano di averci messi tutti insieme a parlare di lui. Ognuno di noi porta dentro di sé un tuo ricordo, Luciano. Il mio ricordo è quello di uno scienziato arguto e brillante, elegante e raffinato, e insieme di un uomo profondo, misurato, garbato, che la sua natura e la storia della sua vita aveva forgiato, dandogli quella profonda umanità che lo ha contraddistinto per tutta la vita.
Okay, so now this is my turn. I will speak in English, both because I want to tell to our guests a few episodes, also because it helps keep my emotion in control. So, as I said before, I joined the Institute in Via San Zeno at the end of 1989. It was me, it was Michela Fagiolini, and Yuri Bozzi. It was a very special time. It was a very special time because we had the privilege of seeing the last rays of a dying sun. Because those that came after us, they cannot even imagine what it meant doing science at that time. And this is something that really forged us. So I will try to explain this to you, to the younger students. How was life in those years? I mean, we were technically very, very limited. We had extraordinary technicians, so Carletto is one who was here, who were building our instruments. So we were not buying instruments, the instruments were built in-house. We were drawing charts on millimeter paper. That is probably something you have never seen. You know, taking notes and then writing down notes again. And we were, especially we, I mean, the youngest, we were at the complete periphery. This was the last province of the empire. We were very far from where action happened. But yet we arrived at a time, that was the time when, no, that was the great idea of Lamberto, that neurotrophins could be mediators of plasticity. And we thought we were, you know, witnessing something that would have changed science. It didn't really, but we were really truly convinced of that. And I mean, what I want to say is that we were asking simple questions with simple methods, and much of the day was spent really discussing. We were spending an extraordinary amount of time discussing among ourselves with the spirit by which you would normally discuss about football matches. So it was not professional, not at all. We were amateurs, completely, you know, deeply in love with what we were doing. And this, I mean, it's also what Nicoletta said. You know, every single question was thought after, every single experiment was really weighted, and we were always discussing what can this mean, what can this implication be. And you know, at that time, for example, also even getting access to paper, it was not that straightforward. So we were receiving once every month the Journal of Neuroscience. It was a great day, and then at some time arrived Neuron. And so, I mean, the level of the discussion was completely different. Discussion was a very important part of our time, and this, of course, is also, or mostly thanks to Lamberto, who always did the most to let us develop our own idea, even if they were stupid, discuss them, always tell what you think, say what you would like to do. And also, as Matthew Diamond said, this was you know, a time when science was not yet part of economy. We were not discussing in terms of deliverables and milestones and Gantt charts and contingency strategies, you know, all these things that we mimic like parrots because they are, you know, drip on top of us from a science that is not ours, that is economy. So it was a very different time, and everything changed in a few years. So I really had the privilege, we had the privilege to still see that type of science. And then I will do a jump of more than 10 years. And this is when I came back from Germany. It was 2000, and I became researcher here at Scuola Normale, and I had this completely crazy idea of studying aging in fish. And there, came out many of the qualities of Luciano you've already heard, and I will not repeat them. But there is one quality that probably has not been spelled out sufficiently, that was generosity. So he really gave me a lab to work on something totally different from what his scientific interests at that moment were. And I'm very proud that he and Antonino, they are on the first papers they did on that topic. And this has really been a turning point in my scientific careers, because probably if he would not have been there, I would have taken a completely different path. Maybe more successful, but maybe not. And then comes the last years. And in the last years, Luciano, he started to write, and he discovered that he was actually gifted as a writer. And so he has left us 2 books. One, which is called Il Vangelo dei Poveri, which is a religious book, where you really can see his religious spirit. And the last one that he completed just maybe a few months before he passed away, that is a textbook on a neural plasticity, which I had the privilege to write the preface, and that was basically, at that time, the only textbook on the topic. For sure in Italian, but I don't know how many are there also in English, specifically on that topic. And that was dedicated especially to the students. This was a textbook written for the students, even if it's written in such a plain way that is also accessible to people who are just curious and educated, but do not have a biological or medical background. And that is another aspect of a man that had so many qualities. And this was really in the last years of his life, he was really more and more into writing science, into making science accessible to people. And I'm sure he also had other projects. He wanted to talk to me and to Yuri about another editorial project that unfortunately he didn't have time to develop. And then the last aspect I want to tell you about Luciano was actually that he was a very, very funny person. I mean, those years that we spent, I remember those years as years where we were laughing constantly all the time. I mean, it was fun. It was not only exciting, it was true, real fun. It was one joke after the other. And so there is one particular memory that really crystallizes and is one of the first time I was perfusing a rat. So he was teaching me how to perfuse the rat. And of course, I mean, you may imagine if you have done it, that, you're really shaky because it's emotionally pretty strong experience. And so I had the rat, and I had to extract the brain from the rat. And so he gave me first the bone scissors, and so opened the skull, and then the skull was open, and there was this brain, and there was a sort of a spoon to spoon the brain out of the brain cage. But I forgot to cut all the nerves that were keeping the brain in place. So I was trying to force it. And at some point, the brain jumped in the air and went directly into the sink and into the siphon. So without even thinking, I jumped below the sink, I opened the siphon, took the brain out, and threw it in paraformaldehyde. And then he looked at me with exactly that same grin and said, "You have to wish yourself that the staining doesn't work. Otherwise, you have to repeat this every single time." And this was Luciano, really. I mean, and I'm telling you one, but there has been a number. Okay, now I would ask Lamberto to tell a few words.
Scusate, non sono venuto stamattina perché ero con degli studenti a Torino al Salone del Libro e mi sono precipitato qui. Ma io ho un particolare ricordo di Luciano perché lui era mio cugino. Io appena laureato a medicina sono stato inviato a prendere la diagnosi. Quindi c'è tutta una storia, insomma, che rimaneva dentro di noi, che è andata avanti per anni. Lui, è vero, ha cominciato con me a fare la ricerca, forse ha imparato qualche cosa da me, questo non lo so, ma è vero quello che diceva Alessandro, che c'era un'atmosfera sportiva, ecco, sportiva. Cercavamo di divertirsi. È vero che eravamo lontani dalle tecniche di avanguardia, come diceva Alessandro, ma non eravamo distanti, a mio parere, rinvagando nel passato, dalle idee di punta. E spesso arrivavamo per primi. La mia idea era quella di fare la ricerca a livello mediterraneo. Siccome quando si mandava i lavori dicevano beh, non ci avete fatto il microscopio con questo, allora tutte queste storie. Poi Luciano ha preso la sua via, diciamo, di ricercatore con successo, ma quello che voglio dire, quindi lui forse ha imparato qualche cosa da me, ma dopo io ho imparato molto da lui. Ho imparato come può essere un uomo, insomma, con tante limitazioni che voi sapete, ma forse io so più di voi. E all'inizio, alla fine, la famiglia, il fratello, tutte queste cose che hanno scompigliato, diciamo, la sua vita. E ho imparato come si può essere coraggiosi, come si può essere, come diceva lui, veramente anche a me mi prendeva sempre in giro quando da vecchio continuava a fare ricerca, mi diceva parole appunto sempre sorridendo e c'aveva questo animo fortissimo, ecco. Lui forse non ha imparato da me, ma io ho imparato molto da lui, cioè come si può essere uomini.
Okay, so I only have two more people on the list, which are Silvia Bisti and Nicola Origlia, but se qualcun altro vuole raccontare qualcosa abbiamo ancora tempo. Altrimenti chiedo di parlare a Silvia Bisti.
Beh, per me parlare di Luciano, e per Luciano è sempre un piacere, perché è una grossa mancanza per me, ma fino a un attimo fa, prima dell'arrivo di Lamberto, io qui ero la persona più vecchia, ero quella che di fatto voi tutti avete conosciuto quando siete arrivati al CNR. Mi ci avete trovato e ora mi ci trovate ancora. Ed è una cosa che non mi piace, perché io ero all'inizio, ma non volevo essere alla fine. Ecco, questo ve lo dico di cuore. Volevo essere che qualcun altro lo dicesse per me. E il mio destino qual è stato? Io ho conosciuto Luciano all'inizio, quando è arrivato, come ha detto Lamberto, ho conosciuto tutti voi quando siete arrivati. E come diceva Alessandro, è verissimo, come diceva la Nicoletta, abbiamo avuto anni bellissimi insieme noi. Ci siamo veramente divertiti, facevamo cose pazzesche con quei tecnici laggiù che ci combinavano di tutti i colori, riuscivamo, in ogni modo ci divertivamo, il plottino. Ti ricordi cosa non abbiamo fatto? Quella carta millimetrata che nessuno di voi conosce più, ma noi le facevamo queste cose. E io ricordo anche un'altra cosa, ricordo quando doveva sposarsi Luciano con Mariolina. La prima volta che l'ho visto ero nella stanza del behavior con la grigia. Facevo behavior ancora, c'erano ancora cose che andavano avanti, in quella stanza lì ho conosciuto lei. E vabbè, poi come ha detto Lamberto, poi ognuno ha seguito la sua strada. Io sono andato professore all'Aquila, Luciano ha fatto il suo percorso e poi la vita ci ha riuniti, perché io ho chiamato Luciano all'Aquila in qualche modo, ci siamo riuniti là e avevamo un progetto ambiziosissimo io e Luciano. Avevamo cominciato il primo anno a organizzare congressi, è venuto Lamberto, è venuto io. Cioè stavamo lavorando bene, ma noi abbiamo avuto un terremoto e questo ha cambiato profondamente la nostra vita, ha cambiato moltissimo tutto quello che avremmo potuto, voluto, dovuto fare. Con tutto questo Luciano non ha smesso mai la ricerca, Luciano ha continuato a Pisa, io ho fatto del mio meglio avanti e indietro, alla fine io e Luciano stavamo insieme in casa, avevamo trovato, perché era l'unico modo per continuare a fare qualche cosa, per essere attivi. Qualcuno di voi lo sa, ci siete stati, lo sapete. È stato uno sforzo grosso, poi io sono venuta via e Luciano doveva continuare quella che era la rinascita. E così non è stato. Cioè io purtroppo mi trovo qui a ricordarlo quando doveva essere lui a ricordare me, dire quello che avremmo voluto, potuto, dovuto fare, non ci siamo riusciti e non ci siamo riusciti fino in fondo. E io vi chiedo una cosa, siccome dell'Aquila qui è presente una sola persona, la persona più giovane che abbiamo, Stefano Di Marco, che mi ha accompagnato, io vi chiederei di fargli dire 2 cose di ricordo di Luciano. Grazie.
Buongiorno a tutti. Appunto, come Silvia diceva, io ho potuto conoscere appunto Luciano quando è giunto all'Aquila, ho avuto purtroppo appunto pochi anni per conoscerlo. Io sono arrivato all'Aquila nel 2015, vabbè, lo conoscevo in realtà già prima, essendo stato già all'Aquila, però dal 2015 ho potuto conoscerlo un po' più da vicino, ecco. E a parte appunto il ricordo professionale che devo dire per me rimarrà un enorme esempio professionale, proprio perché con tutte le difficoltà che c'erano, sia locali che sue, è sempre stato presente comunque all'interno dell'Accademia e all'interno dei nostri laboratori di fisiologia lì all'Aquila, dando un'impronta importante, anche cercando di mantenere una collaborazione, diciamo, all'interno di tutta l'area. Quindi ecco, io il ricordo più grande ovviamente è professionale, perché è stato veramente un esempio e rimarrà per sempre un esempio, ma anche dal punto di vista umano, perché pur essendoci conosciuti poco, per quel poco che ci siamo conosciuti lui è stato in grado di darmi veramente, veramente tantissimo, anche dal punto di vista umano, e quindi niente, volevo solo condividere questo piccolo ricordo di Luciano. Grazie.
Okay, and now the last speech will be by Nicola Origlia, who has or will continue part of the work of Luciano, and he has been his student for all this. You need the? Prendilo il tuo computer. Arrivano, sì.
Ora poi lo manda lei.
Ah.
E con quello si scorre.
Con questo? E per mandarlo? Questo. La prima?
No, lo manda lei.
Ah, ok.
Lo manda lei. Ti vedi?
Okay, I want just to say a few words in English because I want to, first of all, personally thank Andreas for being here and accepting our invitation and because I want to mention something about the link between you and Luciano. A few months ago, I gave a talk in memory of Luciano in a meeting. So that was the opportunity to take an overview at the whole scientific career of Luciano. And I think that a very important turning point was when he went to the United States and visited your lab. Because that was a change, a significant change in his experimental approach. Before that, he was using in vivo model with the very well-known paradigm of monocular deprivation to look at plasticity in the visual cortex. And then he started to work on slices, on visual cortex slices. And this was the approach that he maintained when he was back to Italy, in Trieste, where he set up his own lab that was equipped for doing in vitro electrophysiology. And after a few years is the moment that I joined the lab and started my career in neuroscience doing electrophysiology in visual cortex slices, actually. So I first met Luciano in 2002 because there was an open position in the company developed by a spin-off company of CISA in Trieste, developed by Antonino. So I met first Luciano in CNR, so we started talking, and after a few minutes he stopped me and told me, please, Nicola, call me Luciano. And that was a kind of relief for me. And then for the next 4 or 5 hours, we drove to Trieste, and we discussed about so many things in science. When I get off the car in Trieste, I was thinking to myself, man, you need that job. You cannot miss this opportunity because this is science. So the day after, I met Antonino, and they were so insane to hire me in the company because I had no experience at all in neuroscience. I was completely virgin to that field. So I started my career in neuroscience in CISA, and since then I've been working with Luciano for many years. In Trieste, and then in Pisa, and in L'Aquila also for a period. So I could see all the things that they have mentioned personally. And we had success, failures, as every one of us. And we also faced personal changes in our personal life. And that's what makes two persons really close. So I don't have a special memory because there are so many, of course. So what I can do is to reveal a particular thing about the picture of the. So this is because okay, everyone that I met during these days, they told me, oh, this is a nice picture. This really looks like Luciano. His smile, his face. But it's also true that no one could ever see him wearing ties or in a very smart dressing. So in that day, he was wearing a very nice papillon because that was a really rare event, unexpected event, terrific event. That was the day of my wedding. And I want to show this picture also because Mariolina is here and also Ferdinando. We were all together in that day and we look really happy. Thank you.
This closes the symposium. On behalf of the whole organizing committee, I welcome the guests for coming here a long way and you all for being present. And this is, for us, one way to remember Luciano.